the stage, please. Welcome, sir, please. I request Dr. Oseltamivir, Director, NIPER Authority, to deliver the welcome address.
Good morning to all of you. Today's chief guest, Professor Jairam, and other speakers, Professor Bharatam, other dignitaries attending online, Dr. Narahar Shastri, Dr. Madhavi, and other speakers from various parts of India. Welcome to this second series of this ADDMC, Advanced Drug Design in Medicinal Chemistry 2023. It is the second one in this series. We wanted to go ahead with many of the things in the same line under the same heading of ADDMC 2023, for which you are all cordially welcome, not only to the second event, the future events as well. This NIPER Guwahati was established in 2008. It is the first of its kind in Northeast region under the Department of Pharmaceuticals. Initially with the 3 departments, later on became 8, and this medicinal chemistry was established in 2020. Now after that, we are growing in a progressive direction, especially in the area of drug discovery, both in terms of synthesis as well as computational chemistry. The importance of this particular conference in the drug discovery in NIPERs, especially in NIPER Guwahati, has some special significance. You are aware that Northeast region is blessed with the richest biodiversity. Ever since I joined here, I focused on how best we can bring out that hidden knowledge of the traditional healers who are having the really good molecules and literally treating the various infectious diseases, and they are unable to see the light of the day. How best we can give the scientific support to them through our built-in knowledge. That is my first and foremost thing, and that is yielding slowly results one by one slowly. As of now, we are having almost 75 to 80 traditional healers and 125 compounds of their traditional medicine is being analyzed by our analysis department, even other allied departments like medicinal chemistry. If you talk about the modern drug discovery, it is entirely different from the traditional drug discovery in the form of what you call this computational support. Earlier, whatever we thought about the millions and billions of rupees spending for the drug discovery in the age of not less than 1 decade, and this is a cumbersome. So in order to note what is the structure of the compound in 3D and all, what is its once it's entered into the system, what is its metabolic activity, all the things can be assessed only by computational chemistry. It is not a replacement. I will say it is a supplement in playing the vital role in an integrated program of drug discovery. As I told you that our medicinal chemistry department, ever since it joined, I wanted to classify into synthesis part, industrial relations part, third one is the computational part. Otherwise, we are not having any faculty like IIT's 40 faculty in a department and all. Maximum 4 to 5 faculty if it is a big department. So I have presently only 3 members. I don't want any redundancy among the 3. One should have one separate identical categorical discipline so that they can work together for the common objective of traditional medicine to bring out to the limelight. So one is third part is the computational chemistry. So we are working on the artificial intelligence, machine learning, deep learning, whatever it is there. Frankly speaking, we are talking these words only for the past few years, whereas we are working in this area since ages, but the terminology is different. I think you can see even 1984 itself, people started working on the data mining in the drug discovery. Again, data mining has got the several sub branches, and who are the youngsters, they can take any of the data mining tool which is available freely on the public domain, like a clustering techniques or classification techniques or prioritization techniques. But we never given the terminology of the artificial intelligence at that time, but we are still working. Now that coin has come, the terminology coin is artificial intelligence, deep learning and all. Very happy to tell you, our faculty are very deeply working into the deep learning as well as machine learning. Very happy to share, even I have seen some of the presentations of our PhD scholars. They have applied artificial intelligence techniques even in the pharmacology, even the analysis, pharmacy practice. That means it is a powerful tool. If you use it, it will give you a beautiful results. How to predict all those things in this one based on the past data? That is called pattern recognition. We are all well aware of all those things, but the name has emerged recently. But we have to go along with the evolution to create the revolution. That we are NIPER Guwahati is a very keenly working into this kind of an area for seeing the light of the day tomorrow in the area of drug discovery. Also happy to tell NIPER Guwahati is the second one to have the supercomputing facility, that is Pearl Bioembryo. I will tell a little bit about this particular facility later. But before that, when I was in ACT, it's a coincidence. The same chief guest, Professor Balaram, he was the chief guest of one of my advanced course in bioinformatics, where at ACT, he was a chief guest and delivered the inaugural address of that conference, where we have taken the Pearl connectivity. At that time, my another partner of the program is Dr. Narahari Shastri, who is again one of the chief guests of this function today, who is sitting in Jorhat and attending this function online. So how history repeats. At that time, Param 10,000, the configuration is less. Now we are working on the Param embryo is more. This Param embryo is not a standalone supercomputing facility. I told you it is a second initiative in Northeast to use. The first one is that Param Ishan was established in IIT Guwahati. Later on, it has been upgraded to the recently that was inaugurated by our Honorable President of India and visitor of NIPER, Draupadi Murmu Madam. That is Param Kamrup. That is the super fast computing facility. Whereas now supercomputing facility is a national property waiting for the users. It is increasing its bandwidth, it is increasing the RAM, it is increasing the space left and right, but users are not on par with the development of supercomputing. It can be, so far it is confined only to the a little bit, very few people are working in the drug design, especially through indigenous supercomputing in the drug discovery. People like Shastri, Professor Jairam, Balram, Bharatam and all. So being a tiny institute located in the corner, there is no question of corner. If you start counting from here, I am the number one. It is not overconfident, and other part will be the last one. Only thing, it is a mindset and commitment and a dedication and the contribution that is there in each and every gene of this NIPER faculty as well as students. It is not overconfidence, only confidence. Let me tell about the bioembryo applications in the drug discovery, protein folding, and disease mechanism. Our people are working. It is a 312 teraflops for the machine learning and 150 teraflops for the MD CPU, 1.344 teraflops, and these are all the technical details of the one. You can see, and happy to share. NIPER Guwahati students are working on this bioembryo, and soon they are going to work on the Siddhi, because it is a DST CDAC program. That is a Param Siddhi combination, where artificial intelligence and machine learning programs are ported that can be available to all the people free of cost. And many students are well trained in this, and some of the workshops are also conducted on this supercomputing facility, which is one of the supporting for the drug discovery. We are very honored to have a giant in this field, Professor Jairam, and other dignitaries on this auspicious occasion. Let me welcome all of you to this wonderful session. I hope this 2 days session will yield some of the fruitful results that will be a ray of hope for tomorrow, day after tomorrow, and the years to come in the area of drug discovery. Because if anyone want to talk about any kind of a drug program, drug discovery program from the natural sources, he should be an Indian. The player should be from India. because we have richest biodiversity, as I already told you. Let me congratulate Dr. Vaibhav and his colleagues in the medicinal chemistry department to organize this program. And thanks to the mighty Minister of Electronics and Information Technology for sponsoring this program. Again, in the second time, of course, they have assured me that they will sponsor continuously for this kind of workshops. Again, thank you very much to the mighty, and thank you to all my chief guests and other speakers for coming all the way physically and spending their time to deliver the talk on online. And well, once again, welcome to all my beautiful students as well. Thank you very much. Good day. Jai Hind.
Thank you very much, sir, for highlighting the objectives of today's workshop and also highlighting all the facilities available at NIPER Guwahati regarding the supercomputing facilities. So now it's my immense pleasure to introduce our chief guest, Professor B. Jairam, Emeritus Professor of IIT Delhi. He is a world-renowned expert in the field of protein modeling and structure prediction. Professor Jairam is currently working as a professor in the Department of Chemistry, IIT Delhi, and also a coordinator of supercomputing facility for bioinformatics and computational biology. He developed the gene-to-drug suite for protein structure prediction program, that is Bhagirath, genome analysis program, that is ChemoGenome, and drug designing tool, that is Sanjeevani. So we can see that names itself are fascinating. And he set up a multi-teraflop supercomputing facility for bioinformatics and computational biology at IIT Delhi. And he's known for making the hardware and software freely available at NIPER, at the IIT Delhi website to the global scientific user community. And he served as a member for numerous task force and chairman for many DSC and DBT committees. And he was a vice president of Indian Biophysical Society between 2006 and 2008. And he was an excellent teacher and a mentor and founder for two startup companies named Reinvent, incubated at IIT Delhi from 2006 to 2009, and Novo Informatics under incubation at IIT Delhi from 2011 to 2014. So with this brief introduction, now I request our chief guest of today's function to deliver the inaugural address.
Thank you, Professor Murthy. Greetings to all the dignitaries, Professor Bhartam, Professor Narar Shastri, my good friends, and all other luminaries in the area of drug discovery, my young friends, and Vaibhav. Thank you very much.
Can we have your video, please? Can we have your video, please?
My preview, you mean my presentation?
Video, sir. Your video can be on camera.
Okay.
Yes,
I'm trying to share my screen now. Yeah, a couple of things I would like to say. In fact, I'm a fan myself of integrating Ayurveda with allopathy, especially in the context of modern drug discovery. The idea that we have one molecule and one target, which is conventional to allopathy, versus several molecules and several targets working together to cure diseases, which is the philosophy of Ayurveda. How do we combine these? That has been the inspiration for the later part of my research. But one good news is that the government of India has kindly created the national supercomputing networks across the country, and I'm very pleased to note that NIPER Guwahati is a proud owner of one of the NSM machines. The good news, of course, is that the MIT as well as the DST have commissioned us to put the Sanjeevani software, the drug discovery software that I will share with you, I'll talk to you about today, on the NSM machine located at CDAC. But naturally, this will be available across all the NSM machines. So we are working on it. Most of it is done. Most of the codes are already ported. Compatibility is tested and validation is done. So now it is for the CDAC Pune to allow us to create a front end and also conduct workshops and give access to the users on the NSM machine. So the idea is that the Sanjeevani software, which is developed indigenously, is freely available to all the students and scientific community across the country, so free of charge, as Professor Murthy has already pointed out. So that's the good news part. Now, I'll take a brief historical tour to get to the point where we are today. And of course, I have several names for what I'm doing, integrating chemistry with biology and IT towards a disease-free planet, or you could simply say bioinformatics for a better tomorrow. I'm affiliated to chemistry. and also the School of Biological Sciences. And I keep calling my lab as SCF Bio, which stands for Supercomputing Facility for Bioinformatics and Computation. That's a longer uh, name, and acronym is the SCF Bio. Keep doing it. If you have time, just visit this and you'll find a lot of utilities of value to you in your research. Now, the dream plan that I have is that 10 a.m. the disease is reported, and 5 p.m. the drug is delivered at the doorstep, whatever the disease, right? Is this possible? Well, I believe 20 years from now, this will be a routine matter, whether it's the young brains in the audience who are contributing, or the AI or the ML machines, the robots who are doing it, whoever it may be, but it will happen automatically, right? And how do we proceed? Well, let's create a pathway. If you want to sketch a pathway, to start with a, let's say, genome, and from genome you identify a gene, the rogue gene, and from gene you go to a protein, right? This ATG GCC is a gene that's coding for a protein on the genome, and MAL, let's say single amino acid, methionine, alanine, leucine, tryptophan, etc. Then you create a structure. So what have we done now? From genome, you have done genome annotation, you got the gene, and from gene, you got the protein. This is straightforward, thanks to our Govind Khurana. And from here to 3D structure of the protein. And once you have the structure, then you are unleashing your creativity of structure-based drug discovery to find a small molecule. And using supercomputers or using your software, fantastic software, and ultimately you have a molecule, and the molecule is, let us say, now dispatched to a panel of doctors, and so all the specialists who will decide on what's the dosage, etc. And then it is shipped, the molecule is shipped to the patient. Patient is running, jogging happily, etc. So some such things can happen. Of course, you can always add a lot of detail to the whole story. I'm sure, I hope some one of you will probably do a good job of it. Then the cartoon that I'm present here. So that's my dream plan. And of course, it involves a lot of genomics, proteomics, metabolomics, information technology, and chemistry, since we are dealing with molecules, right? And how do we do it? A few ideas here. When you start looking at why should we be doing drug discovery, and interestingly, 2015 versus 2030, the bacterial and viral infections are coming down, so they are not really on the top, right? And what is unfortunate, however, is that while we are making reports like this, we are not factoring the resistance. Drug resistance is not factored in these numbers, these projections in 2030 and so on. Now, why resistance and how do they work, etc. For those of you who are just beginning, this is for students who are just beginning to do drug discovery. You know, when we say antimicrobial resistance and what are these microbes? And microbial infections could be bacterial, viral, fungal, other parasitic, right? And bacterial infections focusing particularly on the bacteria, pneumonia. I mean, these are some tuberculosis, leprosy, etc., etc. These are some of the common diseases bothering humans. And how do they work? Well, nutrients is one, bacteria take away, or pathogens multiply and rupturing the host cell, or they produce toxins which are deleterious to the functioning metabolism of the humans, and so on. And previously, earlier times when there were no drugs, what would people do? Well, they would sterilize, they use bleach, cooking at a higher temperature. These were but what do we do now? Now we say, okay, create a molecule which will stop bacterial DNA synthesis or bacterial protein synthesis or bacterial cell wall synthesis, etc. This is for the antibacterials. Similar stories for the other diseases as well. Now, what are the drugs which do this job? Targeting DNA synthesis, antibiotics, sulfanilamide, thiamethoprene, etc. These are very well known. Targeting protein synthesis, streptomycin, doxycycline. Some of you, I'm sure, are well aware of these names, linezolid, and so forth, right? And this is, of course, a very famous example of penicillin, which disrupts the cell wall synthesis, right? And serendipity has caused it. But you don't want all discoveries to be serendipitous, right? You want them to be rational. That's where we are in the rational drug discovery domain, our era today. Now, these are Nobels given for drug discovery from 1939 onwards. So remember, this is Second World War time. People are dying because of bacterial infections, especially soldiers. So naturally, there was a big demand for antibacterials, and Frontosil was one. And similarly, Alexander Fleming and others have created the penicillin molecule. And of course, this lady got the prize for figuring out that the beta-lactam, the penicillin, has this nice, interesting four-member beta-lactam. And now, once you have a structure, you can start making little modifications to make more and more powerful next generation penicillin. That was the idea. She also, of course, discovered, solved the structure of the vitamin B12. She got the Nobel for that. But once you have beta lactam, it was not just penicillin. Now remember, we are still retaining the same thing, but we have a new, more very powerful that is used widely even today, amoxicillin. And cephalosporins and monobactams have come about. These are resistant to beta lactamases, which are just destroying the beta lactam ring. That is bacterial resistance, right? So you're trying to overcome the bacterial resistance. So this is streptomycin, again, 1952 Nobel, and for tuberculosis. And protein synthesis, a closer look. Now, where is this happening? Remember, it's happening in the ribosomal machinery. If you have a ribosomal, a smaller unit and a bigger unit, mRNA is passing through, your tRNA is getting generated, tRNA is passing, entering and leaving, creating a polypeptide chain. Suppose you were to make molecules, again is the ribosomal machinery. In this pocket, then what are you doing? You're stopping the protein synthesis, which is what Venki Ramakrishnan and others have done. So Venki Ramakrishnan at IUNAD, what they did was to create molecules, and that can, of course, they've got this structure, this huge structure, which is a fantastic technological feat. And because it has so many big molecules, so RNA, several mRNA, tRNA, proteins, and so it was just a marvelous piece in structure-based structure refinement or structure resolution. So to get to atomic level resolution of so many million atoms is not an easy matter. But the application is very straightforward. This particular ribosomal bacteria does not share similarity with humans. So humans are safe. You're creating little molecules which will block this protein ribosomal machinery. Humans are safe and bacteria, of course, will die. Right? So, but now you may say, why are we talking about drug discovery in the antibacterial sense as of now? Well, that's because we developed kind of a complacency, some type of smugness that we are superior to bacteria in terms of our intelligence. Obviously, we think, right? So 1986 is when we almost stopped the drug discovery in the antibacterial part. And after that is a big void. So all the known antibacterial that are sold in the market today, well, they're all here and they're all done much before 1986, right? Little derivatives may have occurred, but no new molecule, right? If this is a scenario, and if bacteria develop resistance against these known drugs, then what would happen? Even a fall from a bicycle, well, develops a little scratch, and that scratch could be colony for bacteria, and bacteria could kill people. That is as bad as that, right? So that is what AMR can do. So obviously we need to develop some new molecules and also keep them in store. We don't release them to bacteria. So that is the plan, right? Bacteria becoming smarter, resistant to existing drugs, and naturally we need to worry about this AMR, right? Now, a few words on the viruses. Obviously, bacteria, people do give antibiotics to viral infections, but obviously antibiotics have nothing to do with viruses, right? All of you know, antibiotics don't work on viral infections. So we have to do something different for the viral infections. What do we do? Well, these are some of the viruses that you are, including the coronavirus, right? So we stop the entry of the virus into the host. There is one way to create do drug discovery, or we stop the viral DNA or RNA synthesis. The third part is we stop the release of the viral particles. So there are three mechanisms in which you can do this kind of drug discovery. So this is one of the successful antivirals and involves inhibition of the viral uncoating step. So that is amantadine, the ribavirin. I'm sure all of you recognize that this looks like a nucleoside, right? A little sugar here, then there is a base-like structure. So it is like a mimic. Naturally, you would expect this to be involved in some DNA, RNA synthesis, uh, stopping that, right? Acyclovir is again one popular compound. Obviously, it looks like a base, so viruses can mistake it for AGCT or AGCU and then incorporate it, and then obviously it stops, right? Similarly, the AZT was one popular one for HIV a long time ago. Right, so you can see now the connection between the biochemical pathway as well as the molecule that's coming out, meaning you have gone beyond this idea of serendipity. We say, well, we are now talking of directed drug design. Beyond nucleoside analogs, people were more interested in non-nucleoside transcriptase, etc., and NRTIs, because the reason is simple, that viruses are developing resistance to nucleoside analogs. I'll tell you more about it in a short while. Amprenavir, etc., they're become very popular. Tamiflu, of course, this is again has come into notice recently with various influenza infections. Other classes of drugs would be antimalarials, anti-cancer, and so on and so forth. Now, so these people developed that anti-malarial, I'm sure all of you are aware of, UU2 for artemisinin, this from China, and ivermectin for river blindness, etc. These two, they got the Nobel in 2015. So that was the last Nobel for drug discovery. Starting from 1939 to 2015, we have had several Nobels in drug discovery. Now, people say drug discovery is very expensive despite massive inputs from genomics and proteomics. Uh, it is drying up. No, I'm sure all of you have counted how many molecules are available that you can buy. Now, small molecule databases are increasing, growing. In fact, some companies are really ready to offer you 150 million molecules readily synthesized, are ready to be synthesized and shipped to you. So all you have to ask for that molecule and you'll get it right within whatever time, delivery time, etc. If you're in the US, probably same day. India, maybe it'll take a little more time. 150 million molecules are available, but how many molecules have become drugs in the last 80 years? So this is a story from FDA. Only 2,500. This is not to say that diseases are any less. These are many, many more. But in the last 8 years, this is a story. And we expect accelerated uh, discoveries of drugs, but uh, as the statistics show, in fact, drug discovery pipeline is drying up. Something needs to be done radically, right? And so what did these people tell us? Well, they said, focus on the pathway, physiological, biochemical pathway, or structure. So this has given us, in fact, a foundation for the modern structure-based or rational drug discovery. Right, this was the story of 1988 Nobel. So all these three people have focused, in fact, on a biochemical pathway. Earlier, it was like a hit and trial story. So now, when you say pathway, it could be as complicated. This is just malaria, like a little small organism, mosquito doing the job. And look at all this, just one particular organism and all the genes and genomes, which one to stop? Well, this is where I think a lot of systems biology will play a role along with AI and ML. I believe AI, ML plus systems biology can help us in identifying which one is a good drug target. In fact, we have chosen N-methyltransferase for our anti-malarial activity, which is there for uh, falciparum, but it's not there in humans. But there are other good targets. So the expectation is that if you bring in computers into the drug discovery picture, you can cut down on the cost and time involved in drug discovery, which is rated like 14 years and 2.6. 2.6 has now become $3 billion, by the way. So 3 billion on average per drug, simply unacceptable, right? So we expect that we want to cut down on the time as well as the cost. And we hope that computer interventions, CADD interventions can help this process. So what should computer do? Well, computer should at least sort out. If I give you billions of molecules, computer say, well, these are potential candidates, these are not, and then you can do your experimental assays beyond that point. Now, if you ask me a very simple question, what is a disease due to? Right? Why people fall sick? What could it be? Suppose it was a bacterial infection or a viral infection, etc., or it could be more complicated things like cancer or diabetes and so on. Most of the times, it's a protein that is going crazy. If the protein is uh, overactive, you want an inhibitor to stop it down, right? Like the bacterial proteins getting multiplied and so also viral proteins, etc. And if the protein is underactive, very lazy, then you want an activator as a medicine. So most of the marketed drugs are targeting proteins, and 90% of them are inhibitors. So they are trying to stop. Imagine you want to stop a viral protein or a bacterial protein and so on. So it's on those lines. So why not DNA? Well, DNA also could become, once nanobiotechnology takes shape, in good for drug delivery purposes, can handle these big molecules, which when bind to DNA sequences in a unique manner and also pass through two membranes. Remember, for protein, you have to cross one membrane. For DNA, you have to cross two membranes, right? So you have to go through the penetration part, and also your size of the DNA binding drugs is generally larger than the proteins. Small molecules work well for proteins, but for DNA, you need bigger ones. So nanobiotechnology, I'm sure, will take care of it one of these days. Right. Now coming back to this idea of structure-based drug design. So most of the drug targets happen to be proteins, and that is where the interest is. This is just to convince you again that we did uh, an analysis of the genomes of Plasmodium malaria as well as human. So if you want to choose a sequence for a drug target, which will not bother humans, but we should only bother the malarial genome, what should be the size of the DNA? Well, the size seems to be like 18 base pairs. So if you were to create a molecule that will block 20 base pairs on DNA, then you are making a very unique molecule without any side effects to the humans. Now, covering 20 base pairs, remember DNA, so 3.4 angstrom between base pairs. So if you want to make one turn that is 10 bases, you are doing 34 angstrom. Two turns axially will be 68 angstrom or 6.8 nanometers. That's huge, huge molecule, right? It's like 100 aspirins stitched together to cover the DNA. So that's where I was saying that someday, hopefully, nanobiotechnology can take care of these issues. Right. Back to the dream plan. 10 a.m. this is reported, 5 p.m. drug is delivered by Amazon, Zomato at your doorstep. How does this happen? Well, let's start. So I call this pathway as Dhanvantari pathway, Chem Genome plus Bhagirath plus Sanjeevani. Now, in place of Bhagirath, luckily for the whole world, not only for us, for the whole world, there are new methods such as AlphaFold 2, which have come about. But this is not to say that methods like Bhagirath have no place. Bhagirath, they are physicochemical, whereas AlphaFold kind of methods are ML or AI based. So there is a room for physics and chemistry always, right? So that's where this Bhagirath stands for. So Chem Genome does the genome annotation. So you give a genome, it identifies protein coding regions and genes. So that's this little piece. Once you have that done, writing the mRNA sequence is very easy. AGC in 5 prime 3 prime direction, AGC for AGC, and then instead of T you have uracil, single strand. From single strand to writing protein sequence, again thanks to Hargobind Khurana, we can write this down. After this is a challenge. That is where your 3D structure has comes into play. Now, once you have the 3D structure of the protein, either through X-ray or NMR or methods like Bhagirath, or Rosetta, or AlphaFold, etc., you have the 3D structure. Then you can unleash your creativity of structure-based drug discovery. Now, the Sanjeevani can handle protein as a target, can also handle DNA as a target. Right? That's a little drug molecule that's created here. Now, how does it work? Does it really work at all? This idea of Dhanvantari, people say it. I don't know how many of you recall. So, of course, I joined IIT in 1990, and when people were doing this computational chemistry on on these screens, etc., they used to call this as a TV chemistry. What is a TV chemistry doing here in real chemistry, right? In fact, in one of the proposals that I put up to CSIR, I was asking for some money to develop a free energy methodology. Then the uh, committee members were asking, well, where is the chemistry here? You're doing non-van der Waals kind of interactions. You are creating an inhibitor that is blocking a site, but there is no covalent bond involved here. If there is no covalent bond, there is no chemistry. Luckily, most of the molecules that we buy, most of the drug molecules don't make covalent bonds, right? They just do a non-covalent interactions. And Professor Naresh Shastri, in fact, has written a nice review article some time ago in Accounts of Chemical Research. Now back to Dhanvantari. And how does this work? So my friend in School of Biological Sciences, right in Delhi, he's an expert on HPV, world's expert on HPV, and he was asking me, can you use your uh, computer skills to give me a molecule that I can test in the laboratory against HPV? I asked him, what do you know already about this? He said, this HPV has 4 ORFs, meaning open reading frames, which are coding for a gene, right? From genome, ORF gives you the protein, or a gene, and then the protein. And function of these proteins is also known. Then he further said there are some molecules which are already approved by FDA. FDA stands for Food and Drug Administration of USA. If they put a stamp on your drug, you can sell it anywhere. Of course, equivalent of that in India will be Drug Controller of India. So he does the job, but if FDA uh, so, approval is generally accepted globally. So except that these FDS molecules all look like they're all non-nucleoside analogs. When you see a nucleoside analog, immediately you know this must be involved in RNA synthesis, right? Like, and this is involved in viral polymerase. So these molecules are created to block the polymerase of the virus so that the nucleic acid production stopped of the virus and therefore virus gets cleared. That is the plan. But unfortunately, virus started developing resistance and the polymerase protein started having mutations. They're not able to accommodate these nucleoside analogs anymore. So that is the reason why he said, my friend said, don't focus on polymerase. This is not a good drug target for you. Also, nucleoside analogs are not good. So give me a new molecule, give me a new target. I will test it in the experiment. So I asked him, life is very simple now. Just give me 14 years and $3 billion and I will get you a molecule, right? No, more seriously, I said, give me 3 hours. 3 hours, I'll give you a molecule, right? How do you do this? And in fact, this is a challenge to all the youngsters in the audience. Please, if you have not done it before, I request you to do it because we used to run these training programs at IIT Delhi earlier, 1 week long training programs where students from all over the country in batches of 30, they come, they stay with us for 1 week physically before the pandemic. And then they learn how to do this on their own. So what they do, what the students do is they go to the NCBI, National Center for Biotechnology Information, NCBI website, download the viral genome. In this case, it's HBV. You could have done it for HIV or something else, or HIV too, right? So you download the genome. So these are viral genomes, so they don't occupy too much space on your laptop, so no problem, right? So it is downloaded. Now what do? I go to this chem genome, which is already part of SCA BIOS. These are all free. Whatever I'm saying are freely available from the SCA Bio website. You can try it yourself, right? And this presentation, in fact, I put it already on my website. So if you go to my front page, SEO by front page, this presentation is downloadable. So in case you are missing, you're not able to make notes or something, you can always download it and try it yourself. So now I put this viral genome onto this KM genome 3.0. It identifies 5 protein coding regions. Now any one of the 5 could have been a good drug target because they don't share any similarity with human proteins. But since my friend said don't do polymerase, I had to exclude it. And he was further suggesting why not focus on a surface protein. I said, okay, how does it look like? Well, this is how the surface sequence looks like. Where did it come from, by the way? This came from our genome. Genome to gene, from gene to writing protein is straightforward. And that's what it is. So methionine, glutamate, asparagine, isoleucine, etc. So now what's the next step? So I put this sequence into my Bhagirath program. And what it gives me is a 3D structure. So once I have the 3D structure, I'm looking for binding pockets. Remember, I'm not taking any experimental information right now. I'm doing everything I've been issued. It's just like suddenly a new virus broke out. All I know is genome and I don't know anything else about it, right? How do I go about doing drug discovery? This becomes more and more relevant in the context of outbreaks like SARS-CoV-2. Except that people still don't believe that small molecules can cure SARS-CoV. We still believe in vaccines. The reason is simple: 14 years and $3 billion. Everybody's scared of this. 14 years, 3 billion. So all the experts in the audience, CADD experts, must somehow allay these apprehensions and say that, well, we can do it in a couple of years, and for not for 3 billion, maybe much less, like $3 million. That would be wonderful, right? So okay, back to our strategy. So once I have the structure sequence, I have the structure. Once I have the structure, now I go to Sanjeevani software. So my genome has done the job of identifying genes and my Bhagirath has done the job of structure prediction. Now I go to Sanjeevani. I ask for where are the active sites. I identify 10 binding pockets. In fact, every little crevice is taken care here. Every little dip here is taken care here. But most of the times it so happens, when I say most, 70% of the time, the binding site with the largest volume happens to be the true binding pocket at the functional site. But this is not always true, which is why you want to take all the 10. So why not scan molecules against all the 10? What is the idea? Well, here you're dealing with a viral protein. So you want the viral protein to become dysfunctional. You want to block it. So you want a molecule, small molecule, which will occupy that functional site and then block that protein so that virus cannot function and virus get deactivated or virus gets cleared, right? No multiplication happens. So that's the plan. Now, Since people do a lot of repurposing at this stage, meaning, but remember in repurposing, you have only 2,500 molecules, right? FDA has approved 2,500. So your repurposing basket is very small. Why not utilize the creativity that organic chemists have been displaying so far? Attack all those millions of molecules. Let's just take the help of those millions of molecules that organic chemists have created so far, and then do a scan of those millions of molecules again at the binding sites in search of a drug. Now, how do you do this? Experimentally, this is impossible. You want to buy those millions of compounds, and you have to develop an assay, and then you have to have so many test tubes, remember, and you have to have either spectrophotometer or some such calorimeters, etc., to be able to test those things. Nearly impossible to do experimentally. Instead, what we have in Sanjeevani is a method called RSPD. This for a very rapid scan. So I can scan millions of compounds in a matter of minutes. By the way, those of you who have not done it before, yeah, all you have to do is to just go to this site, SCA by site, and type RSPD and put in your molecule or your target and ask for, give me a short list of some small molecules, right? That'll be wonderful. You can try this. That's RSPD. Sanjeevani now can give me a few molecules, and that is about 150. Now this, so I have done the 150 based on some cutoff, like I have minus 10 kcal as a cutoff. I have some molecules which are binding very weakly, some are very tight, etc. And those tight molecules is what I've taken. Also looking for some diversity in the scaffold that these molecules have chosen. Once I have these 150 molecules coming out of this RSPD, which is happening in matter of minutes, on this SEO biomachinery. Then I go to this atomic level docking scoring where you are turning on your van der Waals interactions, hydrophobicity, solvent effects, and all that stuff, right? Coulomb interactions. That is my parallel docking program, which is done at the level of atomic detail, and and it takes care of this atomic level docking and gives me a list of molecules, about 30 here. And then I take those 30 and then put them through MD simulations. Very short MD, not very long, maybe 10 nanoseconds, 15 nanoseconds kind of MD. And promising molecules maybe push them to 100 nanoseconds. So for 5 molecules. So what do I have finally? I have 5 molecules. And this is done. Now it's not really 3 hours. I would say 3 days maybe. In 3 days from a set of millions of molecules for a particular disease, you are able to get 5 molecules, right? And does it really work, right? This is the computational part. Lo and behold, right? My colleague, Vivek Perumal, he has done the testing, and this testing was done in such a rigorous manner. So the referees won't leave him. He was doing viral clearance. He was showing protein ligand binding. He was showing RNA clearance, all kinds of tests. But luckily, it finally got into scientific awards in 2021. And Ruchika is my student who did this Dhanvantari pathway. Kirutika is his student, and then there are others who have done this plasma resonance Raman plasma to demonstrate that they are indeed binding and so on. And Vivek Permal himself is an expert on HPV. So that's a story. Now, very interestingly, he took this molecule we called it 5 here, and administered it to clinically relevant drug-resistant mutation, hepatitis B virus, for which there is no drug molecule available today in the market. And this particular molecule seems to be working extremely well against this mutant virus as well, drug-resistant virus as well. So he published this separately, and I was asking him, there was no need for putting me as a co-author, me or my student. You could have just acknowledged us because we have already done that job in the past. He was saying, how would I ever have known out of millions of molecules, this particular molecule can block the drug-resistant hepatitis B virus unless there was CADD. So I was telling him this is not credit to me, but credit to CADD. That's where we are today in terms of CADD, success stories, right? So it works, and you have to of course be extremely careful while you the way how you do it and how and what kind of softwares are you using and so on. So I believe micromolar heat compound is a routine matter today. With some medicinal chemistry, Professor Prasad Bharatam will tell you more about it, and also Professor Naresh Shastri and others in the audience, and some toxicology. I think toxicology is not in control as of now, and not fully in control, although we have some ideas on how to handle it. So delivering a nanomolar drug with least which is least toxic to the humans, etc., should be foreseeable, in the near future. So it's my expectation, and some of the youngsters may contribute. Now, summary of this story. So this is genomes to hits. So if you have a petaflop machine and the Sanjeevani is loaded already on the NSM machines, I'm sure you can go from genome or anywhere. You don't need to start from genome all the time. You can start from a gene. You can start from the protein. So if you have the structure, you can start from the structure of the protein anywhere along this. So I'll mention a little bit about the Sanjeevani part now. So that's our uh, nice Sanjeevani front end. This is the second version, Sanjeevani 2.0, and I'll stop in 5-10 minutes. Right. Just to briefly mention, Nidhi was the one who spent a lot of time developing empirical potential functions. Now, why do you need them? Imagine you have to evaluate several millions of poses of a doctor molecule in a protein target. You need a very quick, rapid screening function at what works at the atomic level. Of course, there are other methods, ML, kind of methods too. And so this is a contribution from 1990s. But then when we were publishing it and we were saying that this is very relevant to drug discovery, the referees at that time, they say, oh, this is the best you can do from India. We can do perturbation free energy methodology, blah, 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 etc. I felt very hurt, obviously, right? Being an Indian, right? Thought I, I was the one who developed one of those free energy methodology myself. But just because I've crossed Atlantic, I came here to do this drug discovery doesn't mean that knowledge doesn't belong to me. So I went back where I went to for a sabbatical, and I created methodologies such as molecular mechanics GBSA methods, right? So, such PBSA, GBSA. I didn't do PBSA, but GBSA did a lot. Then when I came back, I asked for Parul. So these are all my PhD students on the left side. So what did they do? So Parul demonstrated that you can use MMGBSA for drug discovery, and then she has worked on HIV and so on. Then another student was involved in the uh, Sanjeevani, the first version with COX-2 inhibitors. Then there was Tarun, who was involved in scoring, rapid scoring. So this empirical function gives you, uh, is this pose good? Is this pose not good? Is it better? Is it worse, etc. But finally, free energy, a number. Is it minus 8 kcal? Can I compare this with the experiment? That's what Tarun did. So what he did was to take the dot structure and then get a free energy like quantity which matches with the experiment. And another student did same thing, saw her the same thing for the DNA part. So she did the docking for the DNA as well as the free energy, binding free energy as associated with the docking, right? And also non-redundant database of small molecules. He was a part of it. Then Tanya was involved in Sanjeevani 2.0 version, which involves active site finder and automated docking. So how does it work? It works on it does 8 dockings in parallel, and each one has 20 processors. So you're talking of 160 processors working simultaneously in one docking. So it's done within 20 minutes, very accurate docking. And we have done a lot of comparison with existing methodologies. Then Gautam did that magic million compound library scanning. Remember, you can scan million compounds within a matter of minutes, and then go back to this PARDoc, etc. Right, and then so also he did transferable charge derivation. Some of you who have done some quantum chemistry know that these organic molecules don't come with uh, clear-cut charges, partial charges. You need them for your Coulomb interactions. So this is a method to assign charges. And then there is a method of SOM, which is sites of metabolism prediction. This is for toxicity. Interestingly, when I look at the number of hits to our Sanjeevani, from global scientific community, a huge number are attracted by this Lipinski. This Lipinski rule, of course, is not our invention. Lipinski has done it, but we have a nice website where you upload your molecule in any format and it immediately gives you an answer whether it's a Lipinski compatible or not. This is very popular. In fact, last night, in fact, somebody from USA was asking, "Can I use your web link in my paper?" I said, "Feel free, right? With this all free anyway." Ashutosh spent a lot of time on antimalarials, including mechanism of action of artemisinin, and also creating some new molecules, trizine type molecules against malaria with the N-methyl transferase as a target. Avilash did something very interesting. He did BiTalk, which is reverse docking, meaning it gives you bioactivity information of organic compounds. So he put all the human proteins, structures are available, some of them are predicted, and also some viral proteins. So you give a molecule, What it does is RSPD or product and eventually gives you, well, this molecule binds very tightly to this particular protein. Now, since we know from KEGG database and other metabolism databases, what the protein does, you know which disease it can cure, right? So if you have a molecule which you think is bioactive, you want to know what is it against, right? So you could use this kind of a program, BiTalk type program. He also did multi-targeted of ligand design, one ligand binding to two targets and so on. And in addition to this, he developed a Sanjeevani app for those people, the border generation people who want to travel in a metro, you want to use your Sanjeevani for drug discovery. So this is available. And then the another student developed next version of BAPL based on machine learning. So she took this Tarun's program and then added ML, etc., to make it superior in terms of predicting the binding affinity. So from a doctor complex, in fact, this is something you can also do. You may be getting a doctor complex from any software. Doesn't have to be Sanjeevani, any software. You can put that to that complex to BAPL or BAPL plus server, and you get a decent binding frequency, which compares quite well with experiment. She also did work on chikungunya virus, ER inhibitors, estrogen receptor inhibitors intercalate for DNA, and then Pradeep did something similar like to RSPD for drug discovery for DNA, and also seeing. Then Ruchika, of course, you have already seen that Dhanvantri thing. And there are a few others who have done, these are all project trainees. In fact, I have more than 100 project students, MSc, MTechs, and BTechs, etc. They were trying to work with these PhD students to validate and test the methodologies and so on. And I have this BIM called Bioactivity Information of Medicinal Plants of India. We already have neem, tulsi, and turmeric. And in fact, one can scan them as a part of this RSPD methodologies to see which molecule from among these three plants. We are now expanding the database with all the bioactivity that's available so that you can not only focus on those 160 million compounds or 150 that zinc and other databases are offering, you can also use the BIMP database, right? Now, quickly on the Sanjeevani pathway, this is our 2006 version. So you have a database or you're sketching a molecule, and then you use drug-like filters. This is where your Lipinski will come in. Then you do geometry optimization, QM, assignment of force field parameters. This is where TPAC M4 kind of things can come in here. Then you do active site finder. This is where that ASF and ADS type of programs do. And then once you have the minimization done, this could be done through your force field like Amber, Romo, Cham, etc. Then you have binding frequency. BAPL can do a good job here. And then MD, we said it's optional, but we almost made it compulsory to take care of flexibility, solvent, temperature, and other issues. Then lead like come. That was 2006 version of the flow chart. And we made it more elaborate by 2012. This was our Sanjeevani 2.0, where we also put in this rapid molecule scanning and so on. And delta G part, how do you calculate delta G from using PBSA, GBSA, or free energy perturbation? Well, this simple thermodynamic cycle, where I was mentioning power contributed to this, so that can be done. And you could also use these methodologies to talk about target versus non-target, right? So is it toxicity because of promiscuity, or is it toxicity because of off-target binding? Those things can be assessed using these computational methods. Now quickly, success stories include anti-cancer molecule. When I say success, I mean computer design molecules synthesized in the chemistry laboratory and tested in a biology laboratory for activity. So three components: the chemistry and biology is included along with the computational part. So this was done in collaboration with Amity School of Biotechnology. My colleague there, Seema Bhatnagar, she did the experimental part, and Anjali Soni, my student, did the computational part. So that was against estrogen receptor. The next story was anti-Alzheimer's. This again is done by my colleagues in Jamia Millia, and uh, Ashutosh Chandili was my PhD student. He was involved in this particular molecule. And the next was anti-malarial, this is NMT. And once again, the experimental part was done in Jamia Millia. And here, the picornavirus antivirals, this was done by the my student Ruchika, and the experiment was done by my colleague Manideepa Banerjee. So she did the experimental part. She was an expert on HAV. What she found was that the molecule is more generic, so it could attack more picornaviruses than just HA alone, right? Later on, Because my other student Anjali did this similar work for chikungunya virus. That's the virology part. And I already mentioned HPV part. We did done one three thing with we've done with Vivek, because we were so excited that one of the molecules that we designed through Sanjeevani, this was done by Gautam in collaboration with Hari Babu from Harvard Medical School. Finally, it went into Nature. So Sanjeevani in Nature in 2016 was an excitement story, right, so for all of us. And of course, we did some repurposing work with NSI, NCA, and several other compounds have come about. That's how this app looks like, Sanjeevani app, developed in 2017. That's how it looks like. So I believe that this T part, if ML and AI can help in the T part, the toxicity part, we could say computer-aided drug discovery is much more in control, and you can easily do this 10 to 5 kind of a 10 a.m. drug is reported. At least lead molecule by 5 p.m. is delivered, right? That's possible. Then making it a drug will be a few more steps, obviously, right? So I mentioned to you chem genome, Bhagirath, for genome annotation, structure prediction, and active site finding, rapid scanning, atomic level docking scoring, and eventually MD and a small molecule, some filters and smaller molecules, hopefully a toxicity filter as well, which is coming up shortly, right? And you could enter this pathway anywhere. You don't have to start from the farthest left, right? So those are the nice people who made it possible. And about 25 PhD students were involved in this whole Dhanvantari activity, right? And it's pretty much indigenous, as you can see, right? Thank you very much for your attention. Visit SEO Bio anytime. Thank you.
Thank you very much, sir, for joining with us and delivering the inaugural address with the entire drug discovery process and including the supercomputing facilities and how they are important in this process, entire process. And they are very inspiring. We can see all the publications, especially in the Nature. It's very inspiring. And to bring it to your notice that, sir, more than 300, or I think now it's going to be almost 400 aspects visited and viewed your presentation both physically and online. Thank you very much, sir, for joining with us once again. And now I'd like to request Dr. Vivesan, the Director of Microbiology, to felicitate virtually our chief guest. And sir, we're going to first it to you, give you the address, please. So you can just show it to Sabir.
Professor Jairam, I will come to you in person in Delhi and then hand over this one to you.
Thank you, sir. Thank you very much.
Thank you very much.
Thank you, sir. Thanks a lot.
Thank you very much for being with us and sparing your valuable time. Thank you.
Thank you.
So thank you, everyone. With this, we come to the end.
Welcome back to the ADDMC second series of workshop. Shastri sir, do we have you online? Yes, sir. Good morning, sir. So we are about to start. I request all the participants to please gather in the hall. So we are once again we are blessed and feel lucky enough to have with us Professor Narhari Shastri, who is the director of the NIST Jorat CSIR Institute. We have a very strong collaboration with them, and we are in continuous touch and learning so many things from them. We had a pleasure to visit NIST last a few months ago, where our team interacted intensively, and I also had the opportunity to get intense training once again from him. So he is a world-renowned expert in the open source drug discovery platform. He is a Bhatnagar awardee in chemical sciences in 2011. He is a national bioscience awardee, DBT in 2009. And there is a long list of awards like BM Birla Award for, in 2001. And without any further delays, because we are facing some delays, I would like to invite Professor Shastri sir to please start his talk. Please, sir, over to you.
Thank you very much, Vaibhav and Professor Murthy Garu and Professor Bharatam, Professor Jairam and all of you. It's a pleasure to participate in this today meeting. And also it's a particularly nice thing for me to talk after my good friend Jairam's talk. Are you able to see the slides?
Not yet, sir.
Not yet. So, Now,
Yes, sir.
okay, thank you very much. So I am very happy and proud to speak after Jairam, who has a great contribution from India to develop the software packages. And today I'm going to talk about the need of these development of open source drug discovery software in particular, and also developing software packages in general. The contents of the talk is about software development and our indigenous attempt to develop this disease-specific web portals, which we call it as molecular property diagnostic suite, and the Galaxy platform and its suitability, and the various modules that are there in MPDS. And if there is a time, I will also try to talk about some of our other recent work in this area and the institutional activities of CSI Analyst. If you look at the community of people who are doing computation, they can be more seen as somebody who looks at supercomputing, high performance computing as the main resources. We grew up in the ages where we used to occupy every single machine with our jobs. And the most important, am I audible? Vaibhav, I am audible, right?
Sir, yes, 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 sir.
Yeah, thank you very much. So the capacity and capability appear to be the most important wherewithal for the people who are doing computation to display their ability to do science. Further, there are also branches like mathematical modeling, statistics, first principles calculations, and simulations, where people are even developing theories, and they probably are not very much into doing computations, but they are very much into theory development and algorithm developments, and then try to see that how these particular procedures can be implemented as software packages. Now we also have software packages, databases, web portals, apps, and IoT-based device development, so on and so forth. But when you look at people who are doing computations in natural sciences in general and drug discovery in particular, we can divide them into different groups. Somebody can just develop theories. One can develop a theory based on the receptors, one can develop theory based on ADMET, or they can develop a theory based on the structure-based approaches or analog-based approaches. And they can also develop methods. Methods which will try to implement your theories. And these two sets of people probably require some sort of a computational platform, but the capacity and capability need not be very high. And there are people who are developing algorithms and convert these methods into programs and softwares, either for academic or for commercial purposes. And we have also application scientists who are supposed to be experts in utilizing the software to get the best results. And by definition, these people should have a very, very good understanding of what is theory and methods and their applicability. But in this process, these application scientists most of the times may not be the ones who are developing either theory or methods or developing a program or a software. But if these application scientists are not really able to find out why the things are coming the way they are coming, if they don't understand the theory and methods behind that, they become like a black box users where they don't know what is happening, why it is coming. So their ability to understand this the results of this computational modeling will be very, very limited. In those cases, they are sort of addicted to the using the program, but if the program is not there, if something is happening, they will be having a lot of difficulty to carry out their research. This is what I call it as a vulnerability. So if you look at any problem that you want to develop, a drug, you want to discover a drug. So drug discovery and drug development are two processes, and you can probably come up with different ways of utilizing our science in the four paradigms, either theory or experiment or modeling or data science. And the most important thing is you need to have a question that which particular molecule is the right molecule for eliciting the optimal biological response. But this is a highly multidisciplinary thing and a large number of factors that are there, which probably most of you are aware of it. And the elegance of any particular approach depending on the right question that one asks and get the right answers. You have to ask the right questions and get the right answer for right reasons, using the right method. For example, you may get an answer which is right, but the reason is not right, or the method that you are using is not right. So when you use another question, probably you will get a wrong answer. So it's extremely important for us to understand that we have to ask right questions and get right answers for right reasons by utilizing the right methods. For that, there is a lot of expertise that is required, and then we can have problems. And if you look at any theoretical problem, this can be divided as a classification problem or an optimization problem or a prediction problem. And we can try to use theory for that. We can try to use experiment, or we can use modeling, or now the most important thing is we can use data science. Actually, if you look at the modeling, you have a belief that, or you means somebody who is doing modeling, have a belief that why the things are happening the way they are happening. What is the reason? Can we model this? Can we understand that? What is the reason that a particular thing is happening that way? So this is a belief. Whereas if you go to data science, we are learning from things that happened the way they happened. Many things are happening and we are getting the data. And from there, we are trying to learn. We are trying to understand. So there is a lot of difference between the third and fourth paradigms in science, that is modeling and data science. And then we are using both these approaches now. So if you look at the commercial software or a basic software, or why do we develop software? Maybe you just want to do some basic science, or you want to do some translational, or you want to make some money, commercial, or you just wanted to see what is the utility of this, or can it solve a fundamental problem and contribute to the society? There's a larger purpose to this. So when you look at the commercial softwares, sometimes in the drug discovery field, they tend to be very, very expensive.
Here is internet is there. No?
Hello, you are not able to see. Everything is fine.
Forward the link, we will see in your phone.
Yes, sir, we are able to.
My mail is
Okay. So there is some crosstalk going on. Okay, so the overtly depending on the commercial software in academic training leads to some undesirable long-term challenges. So I don't have to explain this, and the way forward is to see that how do you develop the culture of writing our own codes and share it across the academic community. Otherwise, the academic community, which are using commercial software as a black box, will have a lot of difficulty for the next generation, because no software is permanent. Only methods and theories are slightly more reasonable, but they also will change. And if somebody is trying to focus a lot more on a particular software and then they don't have the idea about why these, what are the methods that are involved and why we are getting these answers for the questions that we ask, we might have a difficulty. So developing softwares are important, and especially in the academic community, developing open source softwares are very, very important. So if you look at drug discovery, there are various approaches that are there. Something based on a disease-oriented approach, or a drug-oriented approach, or a structure-based approach, target-oriented, or an analog-based approach like QSAR and things like that. Whatever approach that you do, firstly, you need to have a molecule which has some hope, that is called hit, then you will have to fulfill some of the conditions for this hit molecule to be called as a lead molecules, like solubility and many other small toxicities and the binding affinity, where you will have a target and things like that. And afterwards, the lead has to be optimized before we can try to take it forward. Because there may be some properties and some properties are not at a desirable level. So you make modifications to the lead and then the final lead will come and then you can subject them to preclinical studies where the drug discovery phase is over and the development phase will start. So we will have to ask different questions and we will have different approaches for understanding why this molecule will fail in the development phase. So drug discovery has a very, very high stakes. If something fails in the clinical stage, this is going to be a bit expensive. But 90% of the molecules will fail in clinical phases itself. And if it fails in the early stage clinical trials, it's fine. But if when it fails in the late stage, there will be lots of problems, and then there are different reasons and ways in which we can predict the failure of the molecules at late stage. And this will be possible utilizing clinical informatics and artificial intelligence and machine learning techniques. So I just wanted to go to the next slide, that our group was interested in developing a package called molecular property diagnostic suite. Professor Bharatam and his team was very strongly involved in this particular activity, which is going on for about 10 years. So the fundamental question that we asked at that time is, as a chemist, you would like to synthesize the right molecule which will be able to elicit the right biological response. So when you have a molecule, what are its properties? How to synthesize these molecules with the desired properties. So that's the reason why we call this as a molecular property diagnostic suite. For example, a chemist will synthesize a molecule and they may not have an idea why they have synthesized and what they are looking at. But if we are able to make a profile of the molecule and see that it will be useful against a particular target, it will be useful as a drug for hypertension or for diabetes or whatever it is, it will be very useful. So this is a question that we wanted to ask, and I'm very pleasantly able to come across the PASS software, because this idea was there in 2004 and I started in 2011. And I was very happy to see that after a few years, I came across the PASS software developed by Moscow group, where the Institute of Biomedical Research Moscow is there. And Professor Vladimir Poraykov is one of the persons who is leading that. And while some ideas similar to that exist, our thing is to develop a software package which can be useful for people to carry out their research in academic institutes in general, and also any other issue. So several groups have come together and about 52 authors have come together and then published a paper in an Indian journal. They are all Indians. So this is the list of authors that are there, and we are very happy about it. And a few papers were published, and things like diabetes, metabolic disorders, they are underway. Diabetes mellitus is already published, and there are several modules which have come in the last few years. So these APDS disease-specific web portals are aimed to gather and collect all the information and utilize all possible resources of the computer-aided drug discovery. So I just wanted to tell you that there is a molecule and you have different ways of representing the molecule, and you have different filters that are there and different approaches that we can use, which are structure-based, analog-based, ADME properties, and many other descriptors, and utilize the machine learning approaches to predict the property of that molecule. So the general architecture of this MPDS, as you can see, you can try to write a program in any language. It may be a script, C, Python, Perl, or any other thing, or even Fortran you can write, and you can even hook some of the executable modules for this workflow. And we are focusing mostly on the chemical space now, and then we have started working on biological space also. And our challenges are lying in the automation, and then we are trying to address that. And there are different modules which are there in the MPDS, like data repository, drug discovery modules, machine learning modules, data analytics, and data format conversion, and so on and so on. So this MPDS is based on the Galaxy, and I will be very happy to see that this Galaxy-based software has two types. One is disease dependent module, and the other class is disease independent module. In the disease independent modules, you will be able to have tools which employ the computational approaches for drug discovery in general. So these tools can be applied for any disease. For disease dependent modules, we will have information which is related to a particular disease. For example, I told you that I have developed MPDS deep tuberculosis, MPDS diabetes mellitus, and MPDS COVID is going on. Actually, there is very, very small difference in the disease independent modules when you take MPDS COVID or MPDS diabetes mellitus or MPDS TB. While there is hardly any comparison in the modules which are involved in the disease dependent. So this is very important for us to realize. And there is a workflow for the analysis of this. You use your data library, and there's a data processing modules, and there are data analysis modules that are there. So these are some of the tools that we have. There are different publicly available tools, and we collect the information from more than 36 different databases, and then try to have a unique MPDS ID. So there are different classification methods, there are different clustering methods that are being done, and we are using also text mining, natural language processing, and there is things like antiviral models, membrane permeability, toxicity prediction, protein ligand interaction analysis, protein interaction analysis. Many of these things were done. And I just wanted to tell you that this is a program which has, for the first time, divided the chemical space based on the structural differences. So now MPDS compound library has more than 147 million compounds, and each of that compound will have a unique ID. The ID, we call it as Aadhaar card, the MPDS ID, which will give you the molecule and its molecular formula, IUPAC name, and it will also give a bunch of properties which can be generated on the fly. So the uniqueness is the MPDS ID will have a way of designating the chemical into a class and a cluster and the serial number. So this is like a very important identifier for that molecule. From this, we will know that to which class it belongs to and which cluster it belongs to. And we can have multiple pages. The second page contains the information of various sources where the compound is taken with some experimental properties. And there can be third, fourth, fifth page. It can have information about that particular molecule in any way that is there. So we, as of now, have a plan to generate the first and second pages of the Aadhaar card. These are to be done on the fly, whereas third, fourth, and fifth pages of the Aadhaar card will be, according to the current plan, will be stored in the external database. So these are the things, and then we also have developed the fragment library, which is published recently, and we are also trying to finish off this MPDS COVID portal. And you can see most of these portals on our website even now. You can have access to the MPDS COVID, MPDS other things, and we also are continually upgrading our other portals like TB, diabetes mellitus, and so on and so forth. So There are custom tools for MPDS. The scripts or programs written for any given problem in CAD and related area can be added to MPDS Galaxy. And this will be available as a tool. So the philosophy of MPDS is you can write any program and you can incorporate in this with very simple command, and it will be able to serve your purpose because this particular program is available to the whole community. And it is inbuilt open source because they don't want to, or the people in Galaxy community, they don't want to really have any hoarding of the information and not disseminating the information. So it is suitable for several drug discovery programs. And we are probably one of the first ones to very, very effectively utilize the Galaxy for the drug discovery. It is essentially integrating various methods like bioinformatics, chemoinformatics, statistics, machine learning, proteomics, next-generation sequencing, computational biology, genomics, computational chemistry, docking, protein-protein interaction, whatever you name it. Our idea is to collect all this information and then make it as a very important platform for data-intensive biomedical research for which the Galaxy has started, and we wanted to have a platform for open source drug discovery research. The main strength of the Galaxy and our reasons to adopt it is a community-based or community-led development. And there are different communities that are there from Australian, Indian, Europe, Netherlands, Czech, France, Japan, so on and so forth. And the core values of Galaxy is the accessibility, reproducibility, and transparency. I give a lot of weight on the transparency because you can really utilize any of the program and you can modify and you can add. And then if you wanted to really put your own tool into this, you can put. And that will be welcome and that will be taken by the Galaxy team. Then such tool will be made available to everyone. So we have done quite a bit of studies in the MPDS chemical space. For want of space, I just wanted to tell you that if somebody is interested, Vaibhav Dikshit and his team also have worked on some collaborating with us. I'll be extremely happy if they want to use, but the software or the web server requires some more time to be very effectively, effective. But I recommend and ask each one of you to go through this MPDS suite of programs that are there in the NIST website, and we will be very happy to get your comments. So we are focusing more on the chemical space and biological space. And after coming from Hyderabad to Jorhat about 4 years back, there is a slight paradigm shift. So I'm focusing more on continuing my work on computer drug discovery, but everything else is sort of new, like software development, AI, ML applications in various disciplines. So we are also looking at various fields like database development, chemoinformatics, machine learning, protein-protein interactions, polypharmacology, SARS-CoV-2, some mathematical modeling, and computational material science is also important. And this is a database which is developed for the entire medicinal plants of the country, which is divided according to states. And this is by far the most comprehensive database, which has more than 22,000 phytochemicals that are depicted with over 6,000 medicinal plants. And we also have this Northeast medicinal plant database, which is also available. And we have the chemoinformatics and machine learning approaches for identifying the antivirals. And this particular feature is now being added to the MPDS. And then people try to see that whether a compound can be having a possibility to be an antiviral or not. And we are also trying to use the machine learning by taking the different snapshots across the molecular dynamics trajectories and then making some sense out of that. And there is a lot of work that is going on in the protein-protein interactions and machine learning again. So I wanted to actually tell you that the things like polypharmacology, network pharmacology are being exclusively used for drug repurposing. And I just wanted to tell you that the group is focusing on utilizing a large number of structure and analog-based approaches along with the machine learning, which appear to give very, very good results in the area of not only drug repurposing, but also in finding out different properties like membrane permeability or the effectiveness and whether they are effective in some other diseases or not. So in the last 4 years, CSIR NIS, we have conducted the summer research training program, which reached more than 16,000 people across the country. And this was a very satisfying thing, which was done with the help of all the staff and students. And we also have done this drug discovery hackathon program, and several of the people who are involved are also a part of it, like Professor U.S.N. Murthy, Professor Bharatam, and I think many people are involved in this particular program. And these lectures, which were given in Drug Discovery Hackathon SRTP, they are available on the YouTube channel that we had. So we also have this COVID-19 testing facility, which was created. And now this is a genomics facility, probably the most functional genomics facility for the entire Northeast. And our institute is also working on Center for Petroleum Research because there is a lot of scope and need for us to work in this. And we also have a awareness center. There are about 4 incubators now, and this will have to increase. And our lab have the NMPB sponsored the Regional Facilitation Center, which will look after all the projects of AYUSH in the country. And this is all possible because very, very talented students are working. And I'm very happy that the Advanced Computing and Data Sciences group, although it's about less than 4 years, it's now very effective, and you can see several people are working. And these are the snapshot of the Advanced Computing and Data Science group. And this is our NIST, and then we will be very happy if some of you are interested to come and then visit. And I thank once again Professor U.S.N. Murthy, Professor Vaibhav Dikshit, and all the organizing committee members of NIPER Guwahati, and also the esteemed speakers that are there, and finally the students who are attending this meeting. Thank you very much.
Thank you very much, sir, for a wonderful talk. We were listening very patiently to all the advice that you have given us. And definitely, I also encourage our own students, NIPER Guwahati, and other participants to go on the NIST Jorat website and look at the work that Professor Sastry and their team has been doing. And we are also collaborating quite actively with that. So this presentation is certainly going to motivate all of us once again. And so our mind is full, but our heart is not full because we don't have you in person here. So next time, maybe you will be kind enough to bless us in person. So, so with this, we once again thank you, and I invite Professor Tamil Vannan to facilitate in silico to Professor Sastry, and please we'll try to send it to you, or we would like if you can visit actually sometime soon.
Thank you very much.
Please,
Sir, are you able to see this?
Thank you.
Yeah, please accept, sir.
Thank you very much. You can give it to the flowers to somebody.
Yeah, sir.
Thank you.
Thank you, sir.
So I asked some of my students to listen to the lectures, and they will be listening. And I look forward for the collaboration with NIPER Guwahati. Thank you.
So with this, we move on to the next Yet another very eminent personality, Dr. Ivan Skutino. We are glad to have him physically here. And so he is a well-renowned expert in QSAR and excellent teacher. He has numerous awards to his credits. Some of those I'm reading only for the interest of time. Professor Khurana Memorial Prize. Yes, so please, sir.
Okay, so I'm the first batsman who's you're seeing physically, right? Everybody you saw what should. So let me thank Professor Vaibhav. Thank you for inviting me, Professor Murthy. Thank you for that invitation to come here to Guwahati. Nice place, a wonderful place. And that's my second visit here. But it's been a long time that I've been here in Guwahati, and I see a big transformation. I think I was here maybe about 15 years back. There's been a tremendous transformation. I saw the airport 15 years back, and I see the airport now. There's radical changes in the airport. And I think there's radical changes also in the state. So that bodes well for you all of you all as students. There's a great future and a great potential for you all at NIPER Guwahati. All the best. Okay. So I'm going to talk about something that we have been developing in the domain of QSAR. First two speakers actually covered the full domain of molecular modeling. Okay, my focus is very small on QSAR. To be more specific, and actually it is QSPR. Okay, and I think all of you all have gone through this learning about what QSAR is all about. So I need to not talk about the basics about QSAR. Okay, the tool that I'm going to talk to you about is actually, you see, the tool and my name are very synonymous. That is because where are my students? Yeah, Anish is here, who was a major development of this tool. So actually, I retired 2 years back. So they thought the last thing that they're going to gift me is a tool that is goes by my name. Okay, so Evans is my name, and the tool that I'm going to talk to you about is also Evans. Evans stands for eigenvalue analysis, incidentally. Okay, but my name doesn't stand for eigenvalue analysis. That's different. Okay, let's go forward. Thank you. No, okay, that's you can all hear me, right? This is a normal class. Okay, fine. Okay, so at Bombay College of Pharmacy, as I told you, our main focus, we did many other things, but our main focus was basically on developing QSAR methodologies. And the online members may not be able to. Oh, sorry. But I can roam around with this, right? I don't need to be confined to one place. I can roam.
Nobody can be confined.
That's much. Okay. So some of the tools that we have developed over several years is basically, one is Korea, and that is QSAR in a receptor-based setting. Okay, so if you have information about your receptor, you can do QSAR in a receptor-based setting. LISAR is basically using principles of similarity to do QSAR. And HOMOSAR is basically an amalgamation of homology modeling, protein modeling, along with QSAR principles out here. Then EQSR is basically a tool of, basically in QSR, you look at a single structure, you compute properties for that single structure, and then you try to do this correlation out here. But you know, actually structures are in dynamic. Okay, there's not a very single structure in solution. And I'm very doubtful that actually there is only a single structure that populates a given receptor or activates a given receptor. I believe there is a set of low energy structures that actually interact with the receptor and activate the receptor out here. So why are we looking at a single structure in QSAR? And then computing descriptors for that single structure. Is it not more meaningful to look at an ensemble of low energy structures and then look at the properties for all of that ensemble and then see how it equates to the activity? That's more meaningful, right? So we thought that actually should be not confining ourselves to single structure and using it for the development of QSAR model out here. Okay, then we had something which we call as occupancy analysis. And this is basically building a three-dimensional space and looking about how molecules populate certain regions of space. Okay, and then bifurcating that space into activity holes, etc. out there. I can't talk too much about this because it's a little bit more intricate to talk about, but that's what they were trying to do. And then the original, QSAR setting, QSAR in a receptor setting, we try to incorporate it with the ensemble approach. So that is the ensemble approach applied to Korea around here. And then we had an amalgamation of Korea along with similarity, and we developed this. And the last one is eigenvalue analysis out here. Now, these were all essentially restricted to pharmacodynamics. If we were thinking about, why should we only restrict our QSAR methodology to pharmacodynamics? There are issues in pharmacokinetics, there are issues in toxicity, there are issues in metabolism. Can we have a software that can address these issues? Why don't we look about activity? Because for a successful drug, activity is just a small block. Okay, many of the drugs fail because of poor admit properties, because of poor admit properties. So let's try to develop a tool that can also now, besides looking at pharmacodynamics, let's look at pharmacokinetics and let's look at admit properties also. Can we develop such a tool? Is there a single tool? I'm talking about a single tool that can address pharmacodynamics, eminent speakers talked about a lot of tools to do all of these things, but that was not a single tool. If you looked at it very carefully, I'm talking about a single tool to address pharmacodynamics, pharmacokinetics, and admit properties like toxicity and barrier penetrations, etc., etc. Okay. So let's see what is this tool. For academic users, they cost in your pocket. Okay. In fact, I don't think any academic users can get access to SimSip and GastroPlus out here. They're very selective. If you ask them, I want to use SimSip and GastroPlus to do pharmacokinetic modeling, they will ask you 100 questions before they ever give you access to it out here. Okay, these tools are just out of academic domain out here. Then there is toxicity, Topcat and Derrick. Many academic institutions can get access to Topcat and Derrick out here. Okay, so you see there are tools to do different things. And if you have a single molecule and then you want to look at its full spectrum, look at its pharmacokinetics and pharmacodynamics and toxicity and admit properties out here, there are the three tools. But the disparate nature of the tools means every time you wanted to do some other properties, you'd have to move into a different domain, into a different modeling environment, re-input all data. Okay. So the disparate nature of these tools means that it is discomforting to use these three tools with one single molecule.
What we are going to do is, why we that
Okay, so there are scratch one, okay, and start doing it. And then if you wanted to predict toxicity, again, You have to go back to page 1 and start inputting new data again and look at toxicity. You know, irritating out there. Can we seamlessly have one software that can help us to do all of these things simultaneously? So if I finish up with the pharmacodynamics, all that I've got to do is basically change my Y data, and I would be able to predict pharmacokinetics. Change my Y data, whether it is toxicity, whether it is admit, whether it is pharmacokinetics, okay, whether it is steady state volume of prediction, whether it is clearance, whether it is T-hub. I just change that and I would be able to do my admit out there. Not easy. We do not allow to do that. Okay, then rather than going back to scratch and trying to do, enter data all once over again right from scratch and irritating out there. So we thought, let's have a tool out there. Okay, and we wanted to work within the well-established QSAR format.
अबे इसमें वो मिलेगा सर्टिफिकेट मिलेगा क्या?
Why use this? Because if you looked at the more messages, docking has given us a lot of tools. Pharmacophore mapping may have given us a lot of success out there. But I think the best of all of these methodologies, molecular modeling methodologies, has been the QSAR formalism out here. So we wanted to develop the tool that is within the QSAR formalism. And now you have QSAR that has gone from different stages, from 2D to the third dimension, to the fourth dimension, to the fifth dimension, and to the sixth dimension out there. And as you go into higher dimensions, from the second dimension to the third dimension and the fourth, a lot of information is being incorporated out here, but the things start getting very, very complicated. Models start getting very, very complicated and very, very messy out here. Okay, so if you noticed, how many of you have even used 4D QSAR? Some of you may have done some modeling with 2D QSAR, at the most done 3D QSAR, COMFAR and COMSIA. How many of you have used 4D QSAR? Very few. 5D QSAR, still few. 6D QSAR, still few. They have not become very to go back and do a drug design is more important out there, right? The models get so complicated that the deconvolution, going back from the model in order to design a new molecule that actually meets those requirements, is not easy. And therefore, you'll see, though 4D QSAR, 5D QSAR, and 6D QSAR have been developed, there have been very few publications over the many years. Okay, so 2D QSAR is the simplest of all. To conceptualize, to think about it, to work with it, it's all simple. There are a lot of drawbacks. Okay, there are a lot of drawbacks. The lack of basically the three-dimensional structure, okay, in 2D QSAR basically is the profound limitation of 2D QSAR. But apart from that, okay, another thing in 2D QSAR is basically it's very difficult to incorporate chirality. Okay, if I have several enantiomers with different activity out there, okay, how do you put chirality in 2D QSAR? So there are limitations, but all in all, the 2D QSAR formalism is the simplest, the most elegant, and the easiest to conceptualize it. And I think deconvolution in a 2D QSAR model is relatively easy. Not all that easy, it's relatively easy, much more easy than, for example, 4D and 5D and 6D QSAR. So we wanted to contain ourselves with using the 2D QSAR formalism in developing a methodology out there. Okay, and as I said, this methodology, we were hoping that whether we will be able to address pharmacodynamics, pharmacokinetics, and admit properties. Let's hope. We have our fingers crossed, and let's see how well this methodology can handle all these disparate attributes in drug design out there. Okay, so the name is called as Evans. It's referred to as eigenvalue analysis. I'll speak about it very briefly out here. And we couple the Evans formalism with modern ML algorithms. Today, the buzzword is to use AI and ML out here. Okay, so we have used ML algorithms in order to develop models for pharmacokinetics, pharmacodynamics, and for admit properties out there. Okay, I want to be very, very brief out here. So what does Evans work around? You need three-dimensional structures. So you can get three-dimensional structures of your molecules from anywhere you want. You want to draw them and use a 2D to 3D software, do that. You want to download them from some website. You want to get them from the internet. You want to get them from the Cambridge crystallographic database. No problem. you have 3D structures, and then you do a little bit of refinement. Okay, 3D structures, you need to do a little bit of cleaning up. You may have to look for the ionization states. You have to look for conformers. All that thing which is very standard in molecular modeling needs to be done. And you can run a very short molecular dynamics simulation in order to get the lowest energy structure out there. Okay, so we do a very short molecular dynamics simulation, and we pick up the lowest energy structure out here. Okay, now just look at it. Now, when we are looking at for example, at pharmacokinetics, trying to study pharmacokinetics, you look at all the molecules that are there in the pharmacokinetic database. The sizes are range from very small molecules with a few number of atoms to molecules with huge number of atoms out there. Okay, so you have this pharmacokinetic data set which is so diverse. Okay, in chemical space out here, that for our methodology, we need a unique numbering method. We need a unique numbering method for all, whatever is the chemical class, whatever is the size of that molecule. Can we find a unique numbering method out here? And we looked at literature and we found that, okay, quite some time ago, there was a Morgan algorithm that helps us to uniquely, number these atoms. What is atom number 1, what atom number 2, atom number 3, atom number 4, atom number 5. Okay, for all the molecules, whatever is size, whatever is its chemical structure, the Morgan algorithm will help you to uniquely give it a number. Okay, start. What is atom number 1? Which atom should be atom at number 1? Which should be 2 and 3? This is not the numbering. Okay, I'm not going to talk about the Morgan algorithm. Those of you who are interested can look up the Morgan algorithm, which is very easy to implement. Okay, that gives you a very unique numbering for the atoms in your molecule. Okay, and I repeat this: whatever is the size of the molecule, whatever is the chemical space of the molecule, your chemical diversity, your chemical structure, the Morgan algorithm will give you a consistent numbering for your molecule, and that is very important for us, that we have a consistent numbering method for our molecule out there. Okay, so once we have done the consistent numbering molecule, then what we do? Okay, so we have numbered our molecules out here. Then I have this matrix, and I then compute interatomic distances out here. Okay, now we have slightly not done very straightforward. We compute the centroid, and from the centroid we actually use to compute interatomic distances out here. But let me say it very simply out here. Okay, so these are the atoms numbered in your molecule. Here also are the atoms which are numbered in your molecule, and this part of the diagonal. This part below the diagonal and this part above the diagonal are these interatomic distances. For example, okay, the distance between 1 and 1 obviously is going to be 0. So in the initial, this diagonal has all 0 values. Okay, so this is the distance between atom number 1 and atom number 10. This is the distance 1.58 angstroms out. So we can get all these interatomic distances and populate the upper part over the diagonal and the lower part below the diagonal. You can populate with these interatomic distances out here. Okay, then what we do on the diagonal, the interatomic distances for the atom to itself is obviously going to be 0 out here. So on this diagonal, we populate this matrix with a lot number of physicochemical descriptors. So we fill in, for example, the geometric descriptors, steric descriptors, electronic descriptors, topological descriptors, thermodynamic descriptors, spatial descriptors, E-state descriptors, and information content descriptors out here. So you can actually build a huge number of these matrices. So in the first matrices, you put, for example, let's say log P, and you have one matrix. Okay, this is for one molecule. Then what I will do in the second matrix, I will replace it with some other properties. Maybe the HOMO energies. Okay, maybe polarizability. Or you fill your polarizability. Okay, atomic polarizability. You can fill out that. So you can put atomic descriptors or molecular descriptors. Okay, as you can see here, this value is consistent, so it's a molecular descriptor. But you can also have atomic descriptors. Okay, you know that you can calculate atomic descriptors and whole molecule descriptors out here. So what I'm trying to say is you can generate for one molecule a huge number of these matrices, which we called it as PD matrices. So what are these matrices? Okay, they contain interatomic distances, which are actually mapping the structure, the three-dimensional structure of your molecule out here. Okay, and then you also have on the diagonal the properties. Okay, and you can have a huge number of all these properties out here. So you can generate 1,000 or 2,000. Okay, as many number of physicochemical properties you want to do, as many number of these properties you can calculate for one molecule. A huge number of this PD descriptors out here. But now comes the question of chirality. For chirality, how do we incorporate in this matrix chirality information out here. So let's look at a molecule basically which has two chiral centers, because for one chiral center, you will not have any differences in the interatomic distance. For more than two chiral centers, two and more chiral centers out here, you construct all the enantiomeric forms. Okay, you construct all the enantiomeric forms, you refine their structures, you do molecular dynamic simulations out here. And for all these enantiomers, you compute all the interatomic distances out there. Okay, and then you see now, okay, in which enantiomer, for example, let's say atom number 1 and 10. I look at all the enantiomers, the distances of atom number 1 and 10 for all the enantiomers, and I see in which enantiomer this distance 1 and 10 is the highest. So what I will do here, for atom 1 and 10, I will populate this cell with the largest distance between all the enantiomers. Let's say you have constructed 8 enantiomers. Out of the 6th enantiomer has the largest distance between atom number 1 and 10, and you fill that here up here. Okay. Now, then you look at that enantiomer. Let's say enantiomer number 3 has the shortest distance between atoms number 1 and 10. You fill it up here out there. So now what you can do now, okay, for chiral molecules with 2 or more chiral centers out here, this upper distance and this lower distance are actually populated from distances from all these enantiomers, the highest distance and the lowest enantiomer. So indirectly, I'm actually taking chirality into consideration out here. Okay, so now we can see now these matrices are going to get very, very complex. There are huge number of such matrices. Let's say I have this matrix now out here. Okay, so what I can do now to this matrix, I can calculate the variance and the covariance matrix, and then I do a diagonalization. You all know this mathematics. You do a diagonalization and you get the eigenvalues and the eigenvectors. Okay, you have done a little bit of mathematics and you know about what are these eigenvalues and eigenvectors out here. So for each matrix, I can get a set of eigenvalues and eigenvectors. At present time, we have kept aside the eigenvectors. We are going to use them a little later, how to use them in modeling out here. The eigenvalues will be numbers. Some of them will be positive, some of them will be negative out here. And what we can do?
Sharing has to be done.
Yeah, Abhijit, it is coming now.
Please continue.
Thank
Sorry.
you.
Okay, so depending upon the size of this matrix, you get a large number of eigenvalues. We'll keep the eigenvectors aside for the time being. You'll get a large number. So these are basically numbers. Okay, so what we try to do, we take the top 3 eigenvalues for each of these matrices. So this matrix now will have 3 eigenvalues. And you'll have another matrix. In the other matrix, what is going to be the differences? The diagonal is going to have some other property. Maybe it is basic, some thermodynamic properties that you will populate on the diagonal. For that new matrix, you'll have 3 eigenvalues out here. So you can have the huge number of these eigenvalues. So these eigenvalues basically now form the descriptors. Okay, they are descriptors. So these eigenvalues are an amalgamation now of something very, very complicated. They encode information about structure in terms of this interatomic distances, and they encode information about the physicochemical properties of that molecule out here. So they are rich. The eigenvalues and the eigenvectors as a rich source of information out there. Whatever you want is already there. Anything else is missing in this eigenvalues? Structure is there in terms of this interatomic distances. Other properties are all there in the diagonal out here. So they are a sort of hybrid descriptors which are contain a lot of information about every molecule out here. So you do this for all the molecules in your database, okay? And you get these eigenvalues, and then you relate the eigenvalues using some chemokinetic method to the endpoint. It could be the pharmacodynamic endpoint, it could be activity, IC50 values or KDs you want to model, or it could be pharmacokinetics, like for example, volume of distribution, steady state volume of distribution, it could be clearance, it could be T-half, or it could be toxicity endpoints. You can then correlate these eigenvalues to these endpoints. So use your modern ML methods to do this correlation between the eigenvalues and your endpoints out here. Okay, let me quickly go. So we tested it with pharmacodynamic data sets, a large number of pharmacodynamic data sets out here, and it did very, very well. Now, that is not a big thing, I would say, because we have a lot of QSR methodologies which do very well for pharmacodynamic data sets out here. So we did not be very surprised that we had a new tool that was doing well for pharmacodynamics out there. As I say, Many people have developed QSAR techniques to do pharmacodynamics very well, to do very good pharmacodynamics out. Now the greatest test is whether this fuel can do well for pharmacokinetics and for admit properties out here. Okay, so first we had we tried to test our Evans methodology and pharmacokinetics database. This is the biggest test of any method that you develop out here. Because when you look at pharmacodynamic data sets, they are not as homogeneous as, when you look at pharmacokinetic data sets, they're not as homogeneous, they're not as clean as pharmacodynamic data sets. Okay, they are messy, they're dirty, there are missing values, horrible. The pharmacokinetic data sets are really horrible to handle. And if you have a methodology that can do pharmacokinetic predictions out here, hats off to you out here. Okay, we'll come to this inherent problems in pharmacokinetic data sets out here. So we plucked out one of the largest data sets in the public domain to test our Evans methodology. And we modeled two of the well-known pharmacokinetic parameters, volume of distribution, steady state volume of distribution, clearance. And you know, these two things, the volume of distribution and the clearance, are related to the half-life. So we modeled the volume of distribution for these molecules in the data set, clearance and half-life. And we wanted to test on toxicity. Toxicity data sets are quite clean, I would say as clean as pharmacodynamic data sets out here. So here is not a big test of a methodology out here. But nevertheless, as I said, we wanted to put it across various endpoints. We also tried to look at toxicity. Okay, and we looked at molecules that were actually measured against the estrogen receptor out there. Okay, so okay, then the next data set is to look at blood-brain barrier permeability. Okay, and a lot of people have done this right from 1970 onwards. There have been a lot of modeling of BBB, and some very good models have been published. Anyway, we said, let's see how good our model stands against all that has been published out here. Okay, so we looked at a data set of 553 drug-like compounds to predict BBB permeability out here. Then we looked at about how, this is important now, molecules that cross the placenta. This is becoming very important these days out here. So we also did our testing against a data set of 248 drug-like molecules, okay, for placental transfer out here. And then we wanted to look at human skin permeability, and we had a data set of 186 compounds, okay, for human skin permeability out there. Okay, so these are the various things that was done, the various softwares and the various molecular modeling programs, and for doing the chemometrics, we use the R software and we use the Python programs to do all these things. Are all very standard and easily available these days. Okay, and then you need to do a lot of validation out here. Okay, we ran through all the validation routines, the validation matrices to show that our models are reasonable or bad out there. Okay, so first I'm going to talk to you about, let me take up the pharmaco. Before I go to estrogen receptor mode. Okay, look at PK modeling. Okay, so what was the size of it? I've forgotten what was the size of this. Okay, PK. Okay. And you should remember what was the size of the data set for PK modeling?
670.
670, thank you. It was a 670 that. Okay, thank you. Okay, now, okay. So there are two ways in which we can model the 670 molecules. Look at them together, irrespective of the chemical class. Okay, build a model for all these 670 molecules out there. Or we can look at them as chemical classes. We can divide them into acids, into base, into zwitterines, okay, into neutral compounds, and do modeling for subsets of these 670 molecules out here. Okay, why do we do that? So the second one is I'm calling ionization-based models out here. So I've broken my 670 molecules into basically chemical classes out here. Now that's going to be fraught with some problem. You know that when you're doing basically QSAR modeling, the larger number of compounds that you have, you're going to have better models out here. Now 670, if I'm going to fragment it into acids and bases and neutral compounds and zwitterines, now my data sets are going to become smaller, and it's going to be difficult to build very good models out here. Okay, but it is more logical, because what is the problem? You know that basically, you know, your ionization state governs a lot of pharmacokinetics. Okay, governs governs a lot of all your kinetic properties. For example, how much is bound to plasma proteins is highly dependent upon the ionization state of the molecule. How much is cleared? How much of renal clearance? How much of habit is based upon ionization? How much is cleared from the kidneys out here? Okay. So now when we put all these molecules together, irrespective of the chemical classes, we are disregarding their modes of pharmacokinetics. All molecules are put, and we are saying disregard their protein binding, disregard their clearance rates and all of that. That is stupidity. That is stupidity, right? That is really stupidity. So you begin to see that actually the ionization state actually governs pharmacokinetics to a great extent. So it's more logical rather than to take all molecules into a global module, okay, which we know at the onset we are going to fail. Okay, which we know at the onset. It's more logical to break them into ionization states because we know ionization is actually closely related to the pharmacokinetics out there. So we have two approaches out there. Okay, and let's see what happens out there. Now, when you basically look at your 670 molecules and you look at PC1 and PC2, and now you see how they cluster, how they all cluster in the same property space, in the same chemical space out here. So this itself gave us jittery. We are going to have some very big problems. Molecules with diverse chemical classes actually clustered all together in the PC1 and PC2 principal component space out here. Okay, so there's a huge lab in chemical space. Okay, with compounds which have unique elimination and in web of phase out here. Okay, so now here are the models out here. Okay, so very obviously we are not the first to do pharmacokinetic modeling out here. Though my methodology is new, there are a lot of literature reports out here. So I'm comparing our models with literature reports. So in the first row here is a global model where we have taken all 670 molecules together and see how they perform. Okay, and then there are these subsets, okay, which we call as basically local models for the acids, for the bases, for the neutral and the sweeter. So the 670 compounds were broken up into these categories out here. Now, when you look at the standard statistics out here, this is a very busy slide. I know in a few seconds you cannot see what's going to happen. All that I want to say is basically, it's not very good. The picture is not very good because if you look at R squared train, okay, R squared test, you can look at these values. And it is not only that our model doesn't fare very good. You see, literature also has published values which are very close to what we are getting out here. Where is this problem? As I told you, pharmacokinetics modeling is extremely difficult out here. So if you look at all of these values, they are far from what are ideal values out here. When you do pharmacodynamics modeling out here, you can get much better values. It is easy to get good values. Okay, all of these values, the parameters are very good for pharmacodynamics data sets because pharmacodynamic data sets are very homogeneous, very clean out there. Okay, and if you put this against any pharmacokinetic test, people will laugh at you. Okay, but these are now where are the problems in pharmacokinetic modeling out here? Look at it. Let's look at first problem. Let's look at clearance. And then you would look at the clearance values for all of these molecules. Can you look at this distribution? This is a skewed distribution. So your molecules, okay, for clearance in your data set has a very skewed distribution. Look at this. Suddenly you pick down and then you taper down out there. Now what is the fundamental property when you're doing QSAR? QSAR is based on the principle that you have a normal distribution. Is this a normal distribution on the face of this? You can look at it. I can compute skewness and kurtosis and all the. Okay, so here is the problem. The pharmacodynamic data sets has a skewed distribution for clearance out here. So with a sudden skewed distribution and you try to put it into a QSAR model, you're going to be fraught with problems out here. Okay, that's why pharmacokinetic modeling is difficult out here. Look at another problem out here. Okay, if you recall your structures, nicardipine, quercetin, rosuvastatin, desipramine, dextrofrenomine out here. Okay, some of them are acidic and basic out here. Look at these different structures, different properties, and look at their clearance. All same. Molecules with diverse structures and diverse properties, from an acid to a base, but the clearance is the same. So now obviously, if you try to work with this type of data, look at it. There are molecules which are neutral, and the clearance, all the same values. Look at their structures. If you recall your structures from your third and fourth year medicinal chemistry and you look at your property, all of them have 10.96 clearance out here. Such a huge redundancy. Okay, so you see the problem with pharmacodynamics. So when you look at those poor values, you actually must appreciate that it's a struggle to get even these values that are here out there. Okay, and Anish and all have struggled a lot, even to get these best of values. We don't look better after doing a lot of fine tuning of the models and the parameters of the parameter we have got out here. Another thing, okay, look at volume of distribution. Look at this. The molecules and the volume of distribution. This is worse than what is previous. Can you see that? Can you see this? For high values of volume of distribution out there, negligible molecules out here. So if I do not have enough of molecules that actually span the spectrum of the endpoints, okay, your endpoint should have a normal distribution out there. Does it look like a normal distribution curve out here? Look at this horrible curve. So how am I expected to get good things? So there are problems inherent in the pharmacokinetics of these molecules out there. Make it very difficult. That make it very, very difficult to do actually modeling of pharmacokinetics. Look at this volume of distribution. Look at molecules like tefmetazole, C4-titan, cefapyrin, fosfenofrilac. Look at these acidic molecules. All have a volume of distribution of 0.13. Look at this. Okay, molecules with so diverse in structures, but their volume of distributions all turn out to be the same. Okay, and I'll be very well. Okay, let me go to estrogen. I'll take another. Okay, so you begin to see pharmacokinetics. So it's a challenge, and I would throw this challenge to all of you. Take up modeling of pharmacokinetic properties out here, and if you can develop good models, good methodologies for pharmacokinetics, I think you would have done a great service to the pharmacy community out here. Okay, take this up as a challenge out there. Let me develop models for pharmacokinetics. Okay, and then I will talk. Bhartam sir is already here waiting for his lecture, so I'll leave. Okay, let me talk about estrogen receptor modeling. Okay. Okay, so why we want to do modeling for molecules that activate the estrogen receptor out here? Okay, there are a large number of compounds which are capable of disrupting the endocrine system, and these molecules basically act through the estrogen receptor out there. Okay, so it is very crucial in order to and you know the physiological importance of this, right? You know the physiological importance of this. So for any molecule, can we predict what is going to be its activation on the estrogen receptor? That's what we were looking at. So if I have a drug and I want to be able to see how does it affect the estrogen receptor, you know that this is now a mandatory endpoint to be measured, right? A mandatory endpoint by the FDA to be measured, that all your drugs must be shown how they act and behave on the estrogen receptors. Mandatory to supply this information out here. Okay, so we take a data set of 132. Now this is not a very large data set, but then this data set was pruned so that you get very homogeneous, high quality. It's a high quality data set out here. Obviously, there is a much larger data set, more than 132 molecules, but Anish worked very hard in order to prune it and make it a homogeneous set of 132 molecules out here. Okay, so how does it look like? Okay, now estrogen receptor modeling is really not difficult. Okay, this data set is very close to pharmacodynamic data sets in homogeneity, in cleanness, in data values, in their distributions out here. Okay, it's fairly close to that. So we are doing fairly good. Okay, we are doing fairly good. We have R-squared train now 0.84. Okay, I know some of you may say pharmacodynamic data sets give us R-square of 0.9 and higher out there. Okay, but this is not a very large data set. We have only 128 molecules out here. So we are not bad, though we have not reached that, pharmacodynamic test point of 0.9. Then only I will publish a paper. Yeah, we are still lower out there, but other values are good. R square M is 0.6 out here. And we use, the Evans methodology was used along with SVR, okay, SVR regression. Support vector machines, you know that there are two forms. You can do classifications and regressions. We use basically SVR regression, and this is the values out here. Okay, if you do 2D SVR, what do I mean by 2D SVR? Take your hands type of descriptor. Forget all these eigenvalues, and eigenvectors. Just take Hans type of descriptors for the same molecules, okay, for your same 128 molecules, and you want to do SVR regression, okay, then you can see the model here. The model is worse than us. So if you use simple Hans type of descriptors, you all know what a Hans type of descriptors out here. The model doesn't have very good statistics, doesn't fare very, very well. So there is something in our eigenvalues descriptors that are giving us much better statistics out here. Okay, look at it. R square train. Okay, standard Hans type of descriptors gives us only 0.46, while we are up at 0.84. Okay, look at this R square M. Okay, is 0.02 versus R 0.6. So there is something. All that I would like to end up with. There is something in this eigenvalues and eigenvectors that are helping us to model, endpoints little more accurately out there. Okay, so I think I could talk about blood barrier penetration out here. Okay, all of you are students. Anish is out here. He's been working on it for the last 5 years on this. So you can bug him, okay, about blood barrier penetration and all he'll talk to you about. Since Professor Bhartam is here and waiting for his time, I will end up my lecture out here. Okay, all that I want to say is the novelty is with us is basically using eigenvalues, which are a hybrid of structure and physico-chemical properties in order to develop a new type of hybrid descriptors. Okay, and that also we have incorporated chirality information, which is very, very difficult in normal 2D QSER. So all encompassing, it looks like the eigenvalues, a hybrid type of descriptors, will give you better results than standard type of Hans type of descriptors when you're modeling several endpoints. Okay, thank you. Okay, so.
So thank you very much, sir. It is very captivating and capturing talk about. So I'm glad to hear that we now have 2 Evans always will be with us. So that is really wonderful. And also, are there any questions from students? A couple of. He will be around with us for another 2 days, so you can always try to catch him up at the lunch or tea.
I'll be there for 2 days. Anish is there for. I'm not saying. Anish is there for much more longer time. I'll be happy to talk to you if you want to know anything about modeling beyond pharmacodynamics. If you want to model pharmacokinetics and other admit properties, we'll be happy to talk to you. Developing those.
Before moving to the next lecture, I must felicitate Professor Evans. So who better than Professor Bhartam? He's there with us. Yes, he's our guest, but as a part of NIPER family, he's also part of the bigger NIPER family. And so in his presence, I request Professor Bhartam to please felicitate Professor Evans Kudino for this wonderful lecture. So now it is my great pleasure to invite none other than Professor Bhartam. He is the head of the Department of Medicinal Chemistry at NIPER Mohali, and we know him all very well from his work on drug design, molecular modeling, organic synthesis, nitrosamines, and carbenes, and you name it. He is with us, and in the interest of time, there are many awards that I can continue reading. And he is an excellent teacher. I had the good fortune to get trained under him. So it's always a pleasure to listen to him once again. Without any further ado, I invite him, sir, please.
Very well, Professor Kudino has given excellent inputs to us. You have several take-home messages from his lecture. One is that ionic states. We can never forget the ionic states of the molecules. We always are carried away. This is a common mistake that Professor Coutinho told us we should not make such mistakes. And also data integrity, data uniformity, data spread. All the many things are associated with the data. We work with data, but we are not good at it. We say that our Chitragupta is recording our data. If he makes any mistake, we go to Narak instead of going to Swarg. So similarly, we don't want to do similar mistakes in data. Okay, so data drug discovery relates to the accuracy of the data. That Professor Coutinho already explained to us. That is very nice. Now I'm going to go a little more towards organic chemistry. How many of you are informatics people? How many of you are medicinal chemists? I don't know. But I hope my lecture will be more towards organic chemistry in this particular application. Okay, it is about the designing antibiotic agents. Is there a acknowledgement or no? No need. So I am going to talk about the antibiotic agent design. This is one of the recent works in our lab. I am going to discuss the details. See here, we employ computational methods at every stage of the discovery, but we also integrate with organic synthesis and then biology. So that is what is giving full circle to the drug discovery perspective. Okay, let us look at the compounds first. This is the compound I am talking about. Imidazo group and pyridine group. Imidazo is here and pyridine is here. When these two are fused, there is a nitrogen in the middle. That kind of fusing is known as 1,2-A pyridine, imidazo pyridines. Okay, these are having many medicinal applications. Cardiovascular activity, anti-cancer, anti-ulcer, anti-inflammatory, antiviral, antibacterial, like that. There are several already reported therapeutic areas in which this functional group has been reported to be very important. So what we wanted to do, whether we can synthesize this class of compounds. That was first we tried what is already known in the literature. Several methods are already known because these molecules are nothing new about it. But when there are so many methods, at least 8 methods listed in this slide, there are several drawbacks. Expensive metal catalysis is required. Environmental hazards are associated with it. Stoichiometric amount of the base is also required. So likewise, a few limitations are observed. So we found, is there any way of simplifying the reaction? Like I2 here, copper added here, and copper acetate here. Can we identify a better method than these? They're all sufficiently good methods, very good methods. Organic chemists are always on the lookout for improving already existing methods. That's what we did. We found that this reaction can be done in water medium. If you take the catalyst NIS. So this is N-nitrosuccinimide is producing the reaction and producing the product. In water medium at 80 degrees centigrade, 6 hours, we get the product. This is the optimization conditions. This is all very nice data, and this is the most important because the reaction can be carried out in open air also. Initially, they thought that O2 is essential, but then at a later stage, O2 supply is not essential. We can further simplify the reaction just by repeat it in open air. So that means what happened, we can synthesize this class of compounds very easily with various substitutes. And then they are all here, which you can see the various yields. Some yields are very good 90, some yields are 63, like that. There is distribution of it. This is normal organic chemistry. It was good success story. We could publish the paper. We have extended this chemistry to another class of compounds where aldehyde group is inserted as an important addendum. Where chalcone is the starting point, you get this product. So here also we got the good results. That was normal, simple organic chemistry perspective. There is no medicine chemistry till now. There is no computer-aided modeling this till now. Now here modeling comes. How to explain a mechanism? Whenever you do a reaction, there is a mechanism with a reaction. How to explain that? What are the energy values associated with the mechanism? Which part is attacking first? For example, here this is the starting material, one of the starting material. Under normal conditions, we presume that this NH2 is participating in the reaction first. Water is going away from here, but that's not what is happening. This nitrogen, this ring nitrogen is attacking at this center, and this bond formation is the first thing that is happening. How do we know? By doing the modeling, computer-aided drug discovery. Part of it is quantum medicinal chemistry. When we do quantum chemical analysis, you get this. First step is exergonic, second step is endergonic, third is also endergonic, fourth is also endergonic, but the fifth and sixth are exergonic. Overall, the energy release is minus 18.5 kilocalories. For this cyclization step, this is a 3D structure. Professor Coutinho already told us we should think in 3 dimensions. That is where if we go at atomic level and electronic level, we get all these details and we can solve the problems using quantum medicinal chemistry approaches. So we do organic chemistry and we can do quantum chemical analysis, explain the mechanism. This is all good news. This is this much. What is there? What is there for drug discovery? Where is it? Where is medicinal chemistry? Normal. But we continue. What is it? This is what we observed. This is not a very great observation. This is a very organic chemist observed for the last 30 years. Everybody has observed, but they simply kept quiet. They did not notice. What is the observation? This is an aromatic proton. The aromatic proton should appear at 7.6 normally, somewhere close to that. But then their peak position is not 7.6, it's close to 9.6. This is a large deviation. Why is this a large deviation? This was a question. When this question was asked, then the students got scared and he went back and asked, compared all his results. Everywhere this was present. So, okay, there must be some reason. He tried to find a reason. Then we went back to the literature. How many people have synthesized such compounds with indole or imidazole or any other species where there is a carbonyl group here and then there is a proton here and this kind of relation is there? What is the peak position here? All the entire literature is filled with so much data telling that it is always appearing at 9.6. Somewhere between 9.5, 9.6 like that. But nobody has observed it, nobody has paid attention to that. Nobody explained why it is. It was observed and kept quiet, that's all. Reported. Then the answer can be very simple answer. There is CHO hydrogen bond. Okay, that is why it is doing this. This is a simple answer, very happy answer, acceptable. Then next question, little bit extension can be asked. What is that? Is that activating that CH center? Or is it only giving the simple peak change? We consulted many organic chemistry experts. They told that no, it's not doing any activation. There is no activation here, just that there is hydrogen bond here, intramolecular, as well as the peak is like that. I don't think there is this CH bond is probably not so very active. Then we look at entire literature on immunogenicity. Has anybody activated this center? No. That was found that there are a few reactions where this CH has been modified, but not with the help of any of these side directing group, only by using some kind of radical mechanisms. See, sp3, sp2 bonds were generated here, and some 5 papers were published. Then the challenge was given to the PhD student. Can you activate this center? If nobody has reported, let us take that as an opportunity and activate that center and publish it in one more paper. This was a simple objective with which we started our work. Okay, then the student went back and said so much, said that no, sir, it's not possible. We have done it. He tried a few couple of reactions and said no, it is probably not possible. That's why nobody has done. If nobody has done, why we should have worry about it? Let us do something which is easy to do. Then we got that doubt. Why we should do something which is easy to do? We should do something which is, if nobody is able to do, we should try and show it, we can do it. Let us do it. Let us compare with other molecules. What are the nearest neighbors? It is indoles. Indoles are very similar to this, have got similar structure, except that there is a carbon here. So if indoles are very similar, is there any activation at this center reported in indoles? Yes, there are more than 20 research papers in excellent organic letters, JOC, JAX, publications are there. How to activate the CH of the indoles? If indole CH can be activated, why not immunoglobulin CH can be activated? What is the problem? This is the extended question. The one question leads to the next question. Then this similarly many questions came up, and then look at the 3D structure of this and perform the NMR spectrum quantum chemically, calculate the NMR spectrum, and indeed find that it should show the 9.6. The quantum chemistry as well as experiment both are supporting, and they take the challenge to activate that. When this challenge was given to the student to say घबरा गया, why this much problem to me, and in the meantime rescue में corona आ गया, 9 months छुट्टी, sir भूल गए होंगे, so after 9 months, so let us do something else, but sir तो नहीं भूलते, so the moment the student was seen after 9 months, so teacher asked the same question, can you activate that CH center? Then student went back and did work, and then one more time corona आ गया, अच्छा हो गया. This was फिर से 4-5 महीने बाद फिर से same question. That means after 1.5 or after almost 2 years, close to that, question did not go away. So the student said that okay, अब तो काम करना ही पड़ेगा. There is no escape. So what she did, she looked at the indole chemistry and what kind of alkylation reactions are being done. Let me do that. She tried it, and she, I will tell you that. Then she tried on some other indole derivatives. Maybe this hydrogen is required or whatever is required. Maybe can we do arylation? One metal is required. She tried many things. After looking at all the existing literature, tried to repeat this experiment on this imidazopyridines, and she successfully failed. That means this reaction did not go away. It did not progress. Many attempts were made, many failures. So then I told that, okay, let us fail many times. Can we repeat the failure with other examples? Yes. That's what she did. She repeated the failure, and she repeated that every time you try to activate this CH center, this CH center is getting activated. This alkylation will happen at the C2 prime position consistently, systematically on many substrates, but the CH center cannot be activated. Okay, so at least failure is there. We can publish the failures. It's good news. Whenever we have failure, we can publish also. Failure means failure in one side can be taken as a success on the other side. Chit bhi hamara, pat bhi hamara in research. So that's what we do. That's what she did. Performed several control experiments and explained the reaction mechanism and then why it is happening. So we have developed a highly efficient and convenient approach for the regioselective alkylation of 2'2-phenyl imidazopyridine. You see, all these beautiful sentences are only to hide our failure. Okay, so this is how publications are coming, and we also got one more publication on that. Now then she continued to, this question did not go away. Can we activate the C5 center, CH center? So that's what rhodium catalyzed activation was carried out. C5 arylation was done. Now what happened? So this aryl group is here. It is leading to C2 prime activation. This aryl group is still here. This is probably the responsible for the problem that is being created. So get rid of this aryl group and place a methyl group here. So if you place a methyl group, can we do this reaction? Yes, that's what was attempted. And then to synthesize such a compound containing this methyl group, she did lots of work and say this compound was new. And in the case of imadazoles, in the case of indoles, such substituted systems were already known, but in the case of imadazoles that were not known, so the student paid attention to synthesize this starting material. Once the starting material was available, CH activation could be done, and then arylation could be done here. So that means what is causing the problem? This an aryl group here was not allowing the reaction at the C5 center. So she had to play tricks to remove that aryl group and place an alkyl group and continue and support this entire thing with proton NMR C13 HRMS mass. And also thinking in 3 dimensions. Every time, think in 3 dimensions. If you want to succeed in life, we should develop this habit of thinking in 3 dimensions. If we think in 2 dimensions, we will remain 3rd grade scientist. Guaranteed. That's what I have seen. Everybody who has succeeded before me, I found that they had developed the habit of thinking in 3 dimensions. Those who did not develop, they remained. My own PhD at when I joined for PhD, 84 people joined. Out of 84, only 3 people became professors. What happened to other 81? They did not think in 3 dimensions. Okay, so develop that habit every time. It is not difficult. If some computers are available today, a long time ago when I did my work, there were no facilities available. You go to the computer center, just within 5 minutes, spend 5 minutes, you will get the 3D structure of every compound that you are going to use. So can we do this? What are all the methods that are already known? These are all the radical-based methods of sp2, sp3 bond formation. So these were known. So these are not of our interest. We wanted to, what are the current limitations? Radical pathway, specific reaction patterns, partners are required, and sp2, sp3, and likewise listed so many limitations, and then perform this reaction finally with the help of boronic acids, and then this C5 annihilation finally was succeeded. These are the optimization conditions. This is the 3D structure. This is the NMR. Compare the NMRs before and after. You see that 9 point, this particular peak was always appearing after the annihilation. The same disappeared. That means indeed the reaction happened where it is supposed to happen. Several compounds can be generated using this particular pathway. So this was the, now if you can do that, you can get the standard reaction conditions, methanol and 80 degrees centigrade, N2 atmosphere is required. First method, this is a monoarylation at C5 position, sp2 bond, base-free method, no pre-functionalization. All these are the advantages of this method. Several control experiments were done to prove the mechanism. This is the mechanism. First step is exergonic. Second is endergonic, again exergonic. Total overall reaction is 54 kilocalories favorable. That means reaction progresses quite very well. These are the 3D structures or the transition states. Lot many things can be obtained here, and then one can get. That means what I am trying to tell here is that when a problem is given to you by your mentor, there are opportunities to run away. If you don't run away, you will get the results of your choice and you will become successful. If you try to run away, you will always get some result. You will always get some PhD. You will always get some job, but you will not have the satisfaction of solving a problem. Okay, that is what has been done. That is all organic chemistry. Now, where is medicinal chemistry? This is drug discovery things. Okay, now let us talk about the biology. This is an enzyme. This is a very well-known enzyme. We have TSJ protein. It is available in many. It's a protein. It's not an enzyme. This particular protein is available in many organisms. It is causing some trouble. This, if we can stop the, if we can somehow inhibit the activity originating from this FTSJ protein, we can design antibacterial agents. This is the information. Okay, having said that, this is this protein FTZ assembles sweet cells as a dense jet ring. There is a, this is the mechanism how the enzyme activates. This enzyme is monomers. These are all monomers of the enzyme. In the presence of GTP, they form filaments. These filaments can be further aggregated into form bundles in the presence of GPSB. And then this bundling is the one which leads to something called jet ring formation. If that jet ring is formed, then that leads to inactivity of several cells, and thus finally it leads to bacterial growth. So if we can prevent that bacterial growth by preventing the formation of these bundles and forming the jet ring, we can claim that we are successful. So what to do? You go to the literature and find out what is the 3D structure. Yes, no, I told think in 3 dimensions. So this, what is the 3D structure? 3D structure of FTJSZ is not available. For the past more than 20-30 years, people are working on FTJSZ. They were working on 1 dimensions. All biology papers you read, they simply write the name of the drug. They don't even show the 2D structure of the drug. They don't even show the 3D structure of the drug. They simply think that name of the drug only is sufficient. But when we come to molecular modeling, AIDD or ADDMC, when we think about, we need to think in 3D. That is where homology modeling had to be carried out, because the 3D structure was not available. That was done, then molecular docking was done, synthesis was done, and characterization and biological screening was done. This is the pure medicinal chemistry approach, using starting with CADD and again CADD, then organic chemistry, then biology. This is a workflow. That was what was done. Library of compounds were selected, primary screening was done, and then secondary screening, then last many biological tests were done. So what is the homology modeling? Most of you probably are aware of what is homology modeling. It is related to structural bioinformatics topic. We can do homology modeling using by comparing two 3D structures, sequence similarity. Professor Raghav is here, expert on this topic. Sequence similarity, sequence analysis, lots of things, finding the motives, finding the, and finally go to the 3D structure prediction. Once you do that, you have to find out which cavities are available, which cavity is suitable for the docking, which cavity where the binding can lead to inactivity of the protein, and all sorts of things can be done. This is one cavity, it is another cavity here, this one, like that we can do, and then finding, whenever we do modeling, we should not trust it. Very first thing is that whenever you get a result from any experiment, whether it is inside the wet laboratory or the dry laboratory, you should first deal with skepticism. You have to validate it. Validate 1,000 times. Your methodology is acceptable methodology or not. Validation was done using Error plot, verify 3D, ProQ, ProSA. Likewise, there are several things. Ramachandran plot is one of the most important methods that was also utilized. And then this is sequence alignment and details. And this is a 3D structure in color. And then we have to find out which is the domain, where is the cavity where we can dock. After doing all that analysis, you realize that this is the best location where docking should be done. That means it is almost like that. When you enter into the classroom, here already many people are sitting here. Which is the best place to find the place? Sit. You have to find. That is not available. Generally, that those seats close to the door are always occupied. So you have to come inside. And do you want to sit beside the professor? Do you want to sit? You want to sit beside your friend? But that is already occupied. What to do? You have to take decisions every time. This is what was done. After I have done that, which are the amino acids responsible for the interactions, and can I design our compound? Whether the compound that we designed will it okay. Doc already known compounds in the literature, that is arbudol. Arbudol is a very well-known antiviral agent. It is being tried for antibacterial effect also. So we thought that several arbudol analogs can be designed, and they are docked, and after that, getting good result from the docking, we went into the synthesis. How to synthesize? Many of our friends already know that this is imidazopyridine derivative. Do you know how to synthesize many of them already? Just a few minutes ago, I described how to synthesize many of those. Use that method and get that product. And brominated here, you have alkyl group here. I told you this alkyl group is here. Convert it into bromine. Once you have the bromine, you can interact with thiophenols, and then you can have many derivatives of 5. That's what we did. And out of the compounds, out of the total 30 compounds that we synthesized, 10 of them, whichever is showing the best docking score, we have chosen. Then we sent those 10 compounds for biological testing. These are the 10 compounds listed here. This is 0, 1, 2, 3, 4. Compound number 01 has got good score. And this score is comparable to the lead compound, that is vitamin K3. Vitamin K3 is the lead compound. And our compound is also showing similar bonding score. That is good news coming from molecular docking. Then we need good information, good confirmation should come from biology. Primary screening was done. Primary screening is showing that this 86% inhibition is shown at 20 micromolar level. So that was happy. And then is there any other good news? Yes, compound number 10 is also showing there is sufficiently good micromolar activity inhibition, up to 69% inhibition. So, okay, score is also good. That means computational modeling as well as the primary screening are giving comparable results, at least in the case of 2 compounds. If it is okay, take this clue, take these 2 compounds and go ahead to the next level of biology. Because biological screening has to be done many times. So many varieties of biological screening have to be done. So this primary screening showed that this compound is showing good inhibition, this compound is showing good inhibition, all others are useless. So out of 10, at least 2 compounds are showing that is P occasion, and this data was sent to, this was done in IIT Mumbai. Then we did this secondary screening. In the case of secondary screening, we got the news that compound number 10 and the compound number 1 is doing consistently improving results, whereas compound number 10 is showing a hazard result. So suddenly increasing, suddenly decreasing. This kind of inconsistency is not very good in biology. We cannot trust that kind of. So drop compound number 10 and continue to the next level of biology with compound number 1. Because biological experiments are highly costly. We have to eliminate many initial compounds which are not really promising. So that was done, and then again, one more time compared just before finally discarding compound number 10, we reconfirm. In the case of the biology, what is required is this kind of curve is there with at low concentration. With an increase in the concentration, this curve is getting flat. If a flat curve is there, that means my compound is doing the right job. But in the case of compound number 10, with an increase in concentration, there is no flatness. With compound number 1, increase in flatness is happening. So, okay, this is another confirmation. Okay, this compound 10 can be successfully dropped. So, we can continue with compound number 1. So, that is a further experiment were done, consistently significant systematic results are being obtained with compound number 1, and so continue. Find out the using this kind of percentage inhibition curves, we can calculate the MIC values. The MIC value is reasonably good, but not really great. So, yes, at least from computer modeling, we are able to lead to a particular stage where we can come to a compound which is a lead compound. Now, lead optimization can be taken up in the next stage. Further, several biological experiments, whether the jet ring is formed or not formed, all sorts of biology was done, further work was done. This is called the jet ring. Actually, this is the ring that we need to avoid. If this ring is formed, we are in danger. If my compound is helping in not forming this ring, we are successful. This is the observation that was done, and compound number 1 indeed is showing inhibition in the formation of jet ring. Though the activity is not very high, that means compound number 1 can never become drug, will never become drug, but will become the lead compound for the drug discovery. So what is happening from the organic chemistry part, which I discussed in the first 3 sections, we have developed a technology to generate C5 arylated imidazopyridine derivatives, which are not available. Because they are not available, nobody tested them for any biological activity. So that is a technology that is readily available. Now, what is the next step? We have the technology also from biological side that these compounds are lead compounds for new drug discovery. That means these 2 good news are available to us. Now it is time to invest the next maybe 50 lakhs. If with this data, if you go to your director will say, okay, take 50 lakhs. Okay, you have to give confidence, right? Why the director will give you so much money? Until and unless you show him that yes, I am 100% confident, I will be able to do it. That is what is done till now. And director is thinking to, director is probably, and when we asked 50 lakhs, he said, okay, take 70 lakhs. Why not 90 lakhs? Why are you asking only this much? Why don't you ask 1.2 crores? This is what director is asking. Chalo bhai, bahut acha, aage badho. So the question is, now we are ready to synthesize next 3,000 compounds on this series. Because we have our own technology, we have our own lead compound. Nobody else in the world knows it. So, go ahead and do it. That's what is the future perspective. Synthesize series of compounds using the same synthetic route. Design many compounds with the aromatic ring here, which nobody was able to do till now. So we say now that we have, and these are very similar to the already known drug molecules for different activity, but for this activity, we are confident we showed it using biology as well as computational modeling. So whenever you try to do work in conjugation, in collaboration, in support with each other, with reference to computational methods as well as synthetic methods, your bosses will be pleased. And then we are now ready to go ahead. So that's what I will do.
Thank you very much, sir, for a wonderful lecture. And it is clear that they have at NIPER Mohali opened up a new chemical space where more medicinal chemistry exploration will be possible. And definitely we will take inspiration and learn from that aspect. So before we end this session, I must invite Professor Tamil Varnan, who is also our in charge, to felicitate Professor Bhartam.
They may want to doubt our chemistry.
Oh, yeah, please. Any questions? He is going out for a visit.
Couple of questions. So, when they do not have any question, can.
Well, let us give chance to students first.
Yes.
Students first, always. Nobody.
He's going away for a day school visit, but he will be back.
Thank you very much, sir, for your nice presentation. So, I'm having a question regarding that water chemistry that we had said was done.
Right.
So, sir, can you go to the slides?
Yeah.
First part of the presentation.
That is emergence synthesis.
Yes. Sir, did you try any kind of surfactant? Because it has been found out that surfactant is basically required to improve the solubility of organic compounds in water.
So in this example, we did not try that yet. We don't want to try as well. You are welcome.
Yeah. And sir, that NIS that you have used, that is one equivalent, I think. NIS or something. So is it catalytic or?
Well, we tried with both varieties. We have got results in both cases. But now I believe that we can go ahead, since we have sufficient quantity, we are going ahead with this stoichiometric amount, but it is better to use it as catalyst.
Yes, yes.
We have seen that.
Sir, and another thing is that with ruthenium metal, CH activation site is different from your rhodium metal. Right. So have you thought of that difference in the identity of the metal that is going to do something?
Right.
Because I believe that rhodium is in +3 oxidation state,
Right.
if I am correct.
Right.
So in that case, ruthenium is 2 oxidation state.
What is happening is, in the case of this compound, imidazopyridine, N1 center has got a lone pair.
Yes.
Ruthenium or any metal is generally attracted to that center. But what is activity required is that activation of the NH. Metal should go to NH center. Ruthenium or palladium or any other metal was not activating that NH. They are always going towards this N1. So rhodium is going to the NH center. So that is metallic attraction between the lone pairs and the metal, and formation of the 5-membered ring with the metal or the 6-membered ring with the metal. Those things are controlling. We have done quantum chemistry there to explain that, but it's not included. Student is included in the PhD thesis.
Sir, my last question is that the thiol coupling with thiophenol, that aliphatic bromine you have substituted. So when you did the experiment, I mean, students did the experiment. So did they use nitrogen atmosphere or kind of open atmosphere? Because thiol is very prone to undergo oxidation or oxidation.
Yeah, it is nitrogen atmosphere. Then we have, in this work, we have used only simple sodium hydroxide base at work. But later we did work with lithium hydroxide. It was also giving equally good result. But lithium hydroxide water complex was giving even better result. So that is news that is not a part of this, but that is another work we are doing.
Okay, sir, I'm done. Thank you.
Thank you very much. With this, we come to the end of this session, but before we break for lunch, there are. So we close this session, but before we discuss.
Okay,
for
so
lunch, there will
we'll
be-
start now. Am I audible to the who is sitting there? Am I audible? Yes? Okay, fine. Okay, so my name is Dr. Ashish Kanhed and I'm representing here the Solice India Technology Private Limited. The Solice is the platinum partner of the Dassault system. So basically Dassault system is group of 12 different brands, of which BioVia is one brand. And under BioVia, you must be aware about different softwares like Discovery Studio, Material Studio, Pipeline Pilot. Then we have Electronic Lab Notebook, then Solvation Chemistry. All these type of softwares are available under the brand BioVia. And today we are going to discuss about the Discovery Studio under BioVia. So during my presentation, I will go through the different modules present in the BioVia. I'll tell you what these modules can do in brief. And towards the end of the overview, what will we do? We'll discuss the library generation in more detail. And we'll show the demonstration how to use the tools present in the BioVia to generate different libraries, different types of libraries of your own interest, not the random libraries. Okay. So to talk about my company, the Solice is founded in 1990 and headquarters is in Japan. And globally, it is present in Japan, China, India, Europe, and USA. And there are only around 50 plus platinum partners associated with the Dassault system, and we are the one of those proud platinum partner. And in India, we found the Solice technology is founded in 1992. And our headquarters is in Bangalore. Research R&D center is in Pune and Mumbai. Okay, so when we talk about the life sciences, one of the important part is the drug discovery and where molecular modeling techniques are of huge importance. Okay, since morning we are listening different types of techniques and open source softwares and command based softwares. Some are easy to access, some are difficult to operate. Okay, so in this life science section, we provide different types of modules under BioVia Discovery Studio. Generally, in drug discovery process, the important steps are target identification and validation that comes under the target discovery. Then once we have discovered or identified the target, we can use the lead identification technique and lead optimization techniques. Okay, so we provide a variety of the modules under these three sections like the sequence analysis, protein design, simulation, then SAR, homology modeling, X-ray studies, then protein-protein interaction, protein small molecule interactions, then ADMET property analysis, the library generation, and so on and so on. We'll discuss one by one. So when we talk about the scope of Discovery Studio, you can see in this hexagonal diagrams on the right hand side, you can see different small molecule related tools are mentioned. Okay, so these are virtual screening, then lead enumeration, then scaffold doping, QSAR, ADMET, etc. At the center, you can see different biological related tools are mentioned, the X-ray, homology modeling, then quantum mechanical techniques and the biological models as well as small molecule interactions. That means the target ligand interaction tools are also available in the Discovery Studio. So we cover almost all chemoinformatics, bioinformatics, and biological sections under Discovery Studio module or Discovery Studio software, you can say. So first, we'll see how this Discovery Studio software works. Basically, it is a server client architecture where you can install your software on a particular system. You can say it as a server. And by using the client, you can access that software from any other system. So it is server architecture, a server client based architecture. And we provide software on Linux and Windows system. And also we have the web client. So on the web browser, you can access this. So let's see which different tools or techniques available in the BioVia Discovery Studio. Okay. So first section, we can say the sequence analysis and the protein modeling. Here you can use the BLAST and SciBLAST technique. Okay, where BLAST, you must be knowing the BLAST works on the BLOSSOM or such type of technique, and the SciBLAST is basically helps you to identify the alignment of query on different templates or multiple templates. So this is already available in the software. Then you can align the multiple sequences or multiple proteins or multiple peptide or amino acid chains using this software. We have renowned homology modeling technique, that is modular, associated with the BioVia Discovery Studio. We can use you can use that using the graphical user interface here. Okay, and you can perform homology modeling of you can prepare different types of proteins, or you can carry out the mutations, and then you can define your protein models using the homology modeling. And after that, you can perform the antigen antibody interactions. Antibody modeling is there. Loop refinement is possible by using two techniques. One is looper, where you can refine one loop at a time, and in the modeler, you can refine multiple loops at a time. Then side chain refinement, when sometimes the side chains goes out of the conformation or wrong conformation is generated. So the chirotor technique is available, which you can use to correct the side chain. So you can make sure by using these different techniques, you can make sure that your protein is accurate, and that accurate protein will help you to get the better or more accurate drug-like molecules. Okay. Again, you can using BioVia Discovery Studio can perform the transmembrane modeling, where you can define the transmembrane. You can perform the modeling in that. You can align, you can add and change the orientation of the membrane. You can fix the particular part across the membrane, and you can perform specific simulation on the selected or fixed membrane, fixed part of the protein between the membranes. Then apart from these, we have protein-protein docking. Here, protein-protein docking not only means the protein-protein docking, but also protein-peptide docking, protein nucleotides docking, which include protein DNA docking, protein RNA docking, and protein antibody docking as well. So here in Discovery Studio, we have ZDock to perform this type of dockings, and using RDock, you can validate the results. Apart from this, one interesting thing is spatial aggregation propensity is also available in the Discovery Studio. When you perform the therapy, when you synthesize or when you formulate the therapeutic proteins, what happens majority of time, certain surface amino acids of that proteins have the aggregation propensity. Okay, and because of that aggregation propensity in physiological system or in the storage duration, they get aggregates, and their therapeutic, they lose their therapeutic utility. Okay, so using this module here, you can identify which type, which are different amino acids present on the surface of the antibodies or therapeutic proteins, which have the possibility of aggregation. And after identifying those amino acids, you can change those amino acids, and you can modify your structure to get the better stability and subsequently better results in terms of activity as well. Okay. Then the next segment of the Discovery Studio is the simulation and X-ray techniques, where you can perform minimization by using steepest descent, conjugate gradient, smart minimization, or Powell method of minimization. Apart from that, we have cascade of molecular dynamics, where you can perform minimization, then production MD. We are strongly associated. NAMD software is inbuilt in the Discovery Studio. You can use the NAMD for molecular dynamics. Then steered molecular dynamics is also possible. The multi-site lambda dynamics is also possible, where you can perform multiple free energy perturbations and evaluate your complexes. Then
in this Discovery Studio, we also support explicit and implicit solvation models. In explicit solvation model, you can perform the what you can say periodic boundary systems, and you can add different ions. You can decide the concentrations or strength of different ions. And in implicit solvation condition, the continuous solvation system is there. You can perform PBSA, GBSA type of analysis. Then apart from regular analysis of your molecular dynamics like RMSD, dipole moment, radius of gyration, we also support the radial distribution function analysis and the principal component analysis, where in radial distribution component you can find the good interactions or bad interactions, and their distance you can optimize. And there are automated and manual protein preparation tools are also available. You can identify the pK values of these proteins. Then apart from that, we also provide the quantum mechanical molecular mechanics techniques, where you can select particular residues or atoms, and you can perform the quantum mechanical studies on that part using this technique. In X-ray, we can use this X-ray technique for electron density mapping. You can identify which type of or which particular water molecule in the active site is required for activity. You can retain that. You can replace with the ligand molecule, and such type of technique you can perform, as well as you can use this X-ray technique for model refinement and validation. Okay, so the next part, which generally majority of the pharma people are using, is the drug discovery, either by structure-based or ligand-based. So one of the technique is structure-based design. We can say it as a virtual, what we can say it's a virtual screening technique also. Okay, so here we have different input preparation modules where you can prepare your input systems. For example, you can prepare your proteins, genetic material, DNA, RNA. You can prepare your ligands, where you can optimize different properties, or you can find out different properties of your ligand. And apart from that, regular protein and ligand optimization or preparation, we also support to generate the fragment-based libraries. Okay, so this fragment-based library you can generate using specific set of ligands of your choice or generally available ligand set. And here we use the recap technique, that is retrosynthetic technique, to generate the library. And this uses the rule of three, that is standard rule in medicinal chemistry, where the fragment should follow, or the best fragments are those who follows the rule of three. So we provide this condition to generate different types of fragments here. Then we have different docking methods included in the BioVia Discovery Studio, where LibDoc is for virtual screening. You can screen millions of compound, and here hotspot based docking is generated. Polar and non-polar parts or regions of the protein or receptor or target are identified, and depending on that, the conformations are generated. So it is less time consuming but less accurate. So when you have lesser number of molecules, like thousands of molecules, you can use the charm-based C-Docker technique, where the simulated annealing molecular dynamics is performed to generate different types of conformations of the ligand molecules. And this is completely grid-based approach. Apart from that, we also provide the flexible docking, where you can allow certain number of amino acids present in the active site region to be more flexible, and this will help you to improve your accuracy of the molecular docking. So this is LibDoc, how it works on the hotspot generation, which identifies the polar and non-polar hotspots. The C-Docker, which is grid-based generation. And once you identify the ligand conformations in C-Docker, it will help you, it will automatically minimize the generated complex system. Like it goes up to 50 or 500 minimization steps, and it will give you refined structure. Then the next approach is fragment-based design method. Okay, here we start with the probing, the binding site, then adding the fragments, and we can modify the scaffold. So there are different techniques. We'll discuss one by one. So in probing the binding site, we have two techniques, that is MCSS and de novo technique, where the multiple copy simultaneous search, that is MCSS. We allow different fragments to interact in the receptor active site, and we generate the MCSS score. And from that MCSS score, we filter out or we select the specific fragments which shows better interactions. And in continuation of this, we identify the interaction of different fragments with specific receptor site, a particular site in the receptor. And on the basis of the particular interaction, their score, we separate out those fragments and we save these fragments for connection or bridging those fragments to get the novel lead molecule or novel molecule. So we'll see the connection technique in coming slides. Apart from this, we have grow scaffold technique. In grow scaffold technique, you can use different types of reactions. Let's say you have one particular scaffold or core structure or fragment which is showing better interaction in particular region of your receptor or target, where you can use, depending on the nature of the functionality present in the core, you can use different library sets and you can perform reactions on that. Let's say, for example, in your core, you have acid structure. So you can use different amines, a set of different amines, and you can perform that reaction to grow the molecule in the receptor active site. Okay, so here example is given. Let's say we have a set of acids and we have set of amines, and that reaction will lead to amide formation. So on that acid part, you can try various acids, different sets in the acid, and in amine part also you can try different sets. So you can generate a huge library which can show better interaction in the receptor active site. So actually, the grows technique is itself a big tool. So it actually works. First you need to select the point of reaction where you want to carry out the reaction. Then you can use the protein pocket to allow the unfavorable to avoid the unfavorable interaction. When you select the protein pocket, the system will understand, okay, this much region is only available to carry out or use the different fragments. Okay? And on that basis, the system will select different fragments which will not interfere or show unfavorable interaction with the receptor site. Then optionally, you can choose any required interaction. Let's say the new fragment which is coming inside should have pi-pi interaction. If you assign that here, then it will choose those fragments only which can show you that type of interaction. If you want some hydrogen bond interaction, you can suggest that, and that type of fragments only will be tested here to grow. Then optionally, you can choose the flexible residues to perform the flexible docking. Then you need to choose the reaction schemes. Here, a particular set of reaction scheme is available in the BioVR Discovery Studio. But if you find some reaction is not available, but if you want to carry out here, you can simply draw that reaction on the BioVR draw or any other drawing tool, and you can save it in .rxn format. And that reaction in .rxn format, you can submit to this software. It will take that. Okay. The beauty of the BioVR Discovery Studio is it is having bundle of different techniques which provide you the systematic way to perform drug discovery process. But if you want to modify that, the system, the Discovery Studio software allows you to modify some part of the software as per your requirement. Okay, then you need to choose the reagent libraries, and you can perform the sampling, the conformational sampling or full minimization. That is also optional. Okay, once you generate a huge library of molecules, all the molecules may not be useful, or all the molecules may not be good candidate for your target, and you can filter out the molecules, or you can clusterize those molecules using Pareto method. Okay, what you can do here is you can choose certain properties which are essential for your activity. Let's say all the molecular properties are not required for particular activity. That is the general rule of medicinal chemistry. So let's say for CNS target, you need the blood-brain barrier permeation. Then apart from that, the interactions or some other properties are required. So here you can select maximum 5 properties, and simultaneously all the properties will be checked on the particular molecule and clustering is done. So you can, out of thousands or more molecules, you can particularly pick the molecules which are suitable for your target system. Then link method is there when you identify different fragments. Depending on the polar and non-polar nature of the receptor active site, specific linking agent can be, specific linking group can be identified from the fragment library, and that will help you to link those fragments to get fit into the receptor active site. Okay. So till now, what we discussed outward in method, like we were growing from outside and we were moving to the center. And now this new technique, another technique is de novo evolution, which starts from the center point and gets expanded into entire receptor active site. Okay, that is de novo evolution technique. So here you can choose any particular course which you want or which you find suitable. And then at a time, you can use 2 libraries, which acts as which can act at 2 attachment points on your core system. Okay, so on that basis, you can grow on 2 reaction points at a time. Let's say you have a library 1, 2, 3, 4 and another library A, B, C, D. So it will generate all possible combination 1A, 1B, 1C, 2A, 2B, 2C, and huge number of compounds will be generated using this technique, and it will use the receptor active site to grow those molecules so that all the molecules will be having good fit into the receptor active site. Okay, the another technique is replace fragment. Let's say you have identified certain molecule which is very good, which is having very good activity on specific target, but you find it is already reported by some other group. So what you can do is you can replace the core of that structure without affecting the activity part, and this replace fragment can help you to do this. It works on same line to the grow technique, where you need to select the specific region which you want to replace. Then you can use the protein pocket to guide this selection. And optionally, you can choose the specific interaction. Let's say pipeline interactions required or specific other requirement is there. And flexible residues, you need to choose the fragment library, and other steps are same which we have discussed in the flow technique. Okay, so apart from this fragment-based drug design, we support different pharmacophore modeling technique where you can perform structure-based pharmacophore, fragment-based pharmacophore, qualitative pharmacophore, and quantitative pharmacophore. Here, quantitative means you can use that model to extend to QSAR and predict the biological activity. So all these types of models are possible. So these are the conventional pharmacophore model. But what I want to discuss here is ligand profiler, which is also available in the Discovery Studio. So ligand profiler is the very good approach. Let's say if you have a particular number of compounds from natural source or synthetic source, marine source, or any other source, but you don't know on which target it is useful or it can act. Okay, so in that case, what you can do is you can use this ligand profiler technique or ligand profiling technique, and this technique will help you to identify specific or particular target where your molecules can act. Okay, so we have a database of around 253,000 plus pharmacophore on almost 18,000 of protein, and you can find the classification of protein in the software till the PDB level. Okay, and on this ground you can decide your library may be useful for kinases, or your library can act on the particular cancer target, or your library can act on certain CNS disorder. Okay, so this type of predictions can be done, and you can take your research in that direction using those molecules. Okay, and it is available in the Discovery Studio. So this ligand profiler library, which I mentioned, 253,000 plus, it is the world's largest library, and that is available only with the BioVia Discovery Studio. Pharmacophore library. Okay, so now we will discuss this library design and analysis. So we'll discuss some parts in detail and we'll see the demonstration. Okay, when you see the drug discovery process, if you are aware about, if you know the protein structure and ligand structure, we go for structure-based drug design. If protein structure is not known and ligand structures are not known, then we go for QSAR. And if the protein structure is known, ligand structure is not known, we go for de novo design. But in all these three approaches, we require the correct or right library. Okay, if we use the correct library, then only we can land into the right place. Otherwise, it may lead you to the wrong molecules. Okay, so in this BioVia Discovery Studio, we have library enumeration technique where you can generate a library on the basis of reactions, on the basis of markers. Then we have a novel ligand generation technique, which is based on the breed technology. We'll discuss what is that. Then you can generate the fragment library using the recap technique. Then once you generate different libraries as per your need, you can calculate their properties on the basis of ADMET. You can calculate 1D, 2D, 3D molecular properties, including the fingerprintings, where we have atom type, extended connectivity fingerprints, and functional class extended connectivity fingerprint. So this is patented technology by the BioVia, and it is available only in the BioVia, which you can use for different types of calculations. Like you can use this for QSAR or other property clustering or property filtering. Okay. So apart from that, we have similarity selection techniques, multi-objective Pareto optimization technique. Okay, so we'll discuss the library enumeration and novel ligand generation using breed technique. Okay, so library enumeration based on the reactions. Let's say you are performing particular reaction in your laboratory, and instead of synthesizing all possible molecules in the laboratory, you can generate that library virtually in your system, and you can perform or you can calculate different properties on that. And depending on the properties, which is the suitable property or acceptable property range, you can synthesize only those molecules in your laboratory. So here you can draw a particular reaction on BioVia Draw or any other drawing tool, and you can save in .rxn format and you can submit it. We'll see in the demo how to do that. Okay. Right. Then we have library enumeration by markers-based technique. So here we require a core molecule then we require list of attachment points. Okay. Once you have a core molecule and list of attachment points, then you can assign particular fragment set to the particular attachment point, and that attachment point is defined with particular atom. Let's say hydroxyl or chlorine or fluorine. You can see on this SMRKS reaction scheme, we have these many types of reactions included in the software. Okay, where the carboxylic acid, primary alcohol, secondary alcohol, halogens, amides, aldehyde, grignard, all these reactions are included. But if you don't find your interested reaction, your reaction of interest in this list, you can add at the bottom of this list, you can include your own reaction, and that will work in the software. So you can modify the software as per your requirement. Okay, so we'll see how this technique is used in the demo session. The novel ligand generation using breed approach. Okay, so when you have two molecules, when you align those two molecules, depending on a particular criteria, it will swap the bonds and it will generate a new breed or new generation. And that combination of molecules will be newer one. Here I have given a small example, but in demo we'll see its utility. Let's say you have a data set for your QSAR and you have aligned those molecules. You can use those 50, 100, 200 molecules here, and you can generate a huge library. Okay, here we can go for 1 generation, 2 generation, 3 generation, up to 6 generation. But as per observation, 3rd generation will give you maximum result, and after that you will find the repetition in the molecules. Okay, so here this breed technique actually works, or it considers 4 different conditions. One is hybridization state, that is nature of bond. Okay, sp3 hybridize will swap with sp3 hybridized system only. It will not swap with the single bond, will not swap with the double bond, like that. Okay, then the distance between 2 points should not be more than 1 angstrom. Okay, so for example, 1 point in a one molecule and second point in second molecule, the distance between those 2 points should not be more than 1 angstrom. Okay, and the angle between two bonds of molecule A and molecule B should not be more than 15 degrees. Okay, why this criteria is decided or restricted here? When you see a particular molecule in the receptor active site, okay, when you replace it, when you replace particular part of the molecule with another part, okay, what happens if that replaced part is occupying the same receptor region? Then you will get similar type of possibility, plus possibility of getting similar type of interactions will be more. So that's why these restrictions are mentioned over here, so that you will get more similar type of molecule. Okay. Okay, and the ring systems, the bonds present in the ring systems will not be replaced or swapped. Okay. So we'll see, you can see here the 2 molecules are mentioned. These are aligned and you got different molecules. So from these 2 molecules, you got 4 molecules in the first generation, like this. We'll see in demonstration. So if you have a particular QSAR data set, you can perform that. But the beauty of this technique is, if you have a particular disease condition known to you, for example, let's say anticoagulation, where factor 10A performs a major role. So in that, what you can do, you can download all possible or all available protein data bank structures. Okay, the proteins are same and the ligands present in the active site are different. But when you align all those proteins, you will get active site based alignment of diverse molecules. Okay, and you can use that aligned molecules, ligand molecules, in this technique. And we have used such type of technique and in the factor 10 only, and we got a very good result of the molecules, which is we found one molecule which is more active than the apixaban, which is the marketed molecule. Okay, so such type of techniques you can use. You need to apply your logic. The software is there, but you need to select the sequence and you need to apply your logic, which can lead you in proper direction. So we'll see how this technique works in demo part. The recap technique is there where fragment library generation is performed. Here we use the retrosynthetic combinatorial analysis procedure, that is recap. Okay, and this is completely, or this is based on the rule of 3, which is the very important rule in the medicinal chemistry. And the breaking of the bond is carried out at particular specific positions, and those positions are mentioned over here. Okay, so now we'll go, or we'll see the demo one by one. We'll discuss these techniques one by one using the software. 15 minutes. Another 15.
Any questions?
Yeah, and till now, if you have any questions, we can discuss.
Don't hesitate. This session is for you only, and all the sessions in fact are for you only. So it doesn't matter if it's a simple question.
कोई भी छोटा बड़ा questions, don't hesitate. And I'm here. Tomorrow also I'm here, so you can personally meet me. Ask me if you don't want to ask here.
May I ask you one question? Yeah. Okay. So here, Dr. Pawan Gupta from SVKM Institute of Pharmacy, Dhule. So actually, it's a very wonderful software that we use actually.
So you have this software at your institute, so you must be aware about the basics.
Yes,
of this
yes,
software.
of course. But
So
I have some
this is Discovery Studio client. And there are different techniques in macromolecules, simulations, receptor ligand interactions, pharmacophore, small molecules, X-ray and MyTools. Where in MyTool you can generate your own sequence and of your choice, and you can use that in single click or step by step. You need not to go here or there for particular module. So you can arrange all required modules in sequence here. You can give a specific name, let's say a heat, or let's say COVID-19, or let's say whatever name of your project, and whatever techniques are required, you can get those techniques in that particular segment.
Hello.
Okay, so we'll discuss the design and analyze library. There we'll discuss enumerate library from ligands.
Hello. Hello. Hello.
Okay. So I'll take this simple molecule here. This is aminophenol, where one hydroxyl group is present and amine group is present. So on hydroxyl group, you can use different acids to perform esterification type of reaction. On amine, you can use different haloalkanes to perform alkylation. So this basic technique we'll use. So when you click on enumerate library from ligands, you can select the, okay, there is only one molecule available, so that is why it is showing same molecule. If there are multiple molecules, you need to navigate to your molecule. Okay. Then R groups. Okay. Now R group is not showing anything because nothing is selected on the visualizer. So for that, let's say hydroxyl H, will select hydroxyl H. That is one reaction center, and hydrogen of NH2, that is one reaction center on which reaction will take place. Okay, using shift key, you can select multiple atoms, then go to enumerate library from ligand. So your molecule is selected. R groups, create new from selection. You have selected two centers. When you click on it, you can see on the screen R1 and R2 is mentioned, right? Then R1, you can import the R1 groups in the visualizer and you can select visible molecules or you can browse your own library. Okay, so I will select for R1, that is amine. So I will select haloalkanes. Okay, I have selected haloalkane file where I have created different chloroalkanes, 8 to 10, I think 8 to 10 chloroalkanes are there. Now in attachment point, you have to mention, clearly mention which type of atom is present in your library. So I will select chloro. I'll show you that library first. Right. So this is one of the molecules from the R1 library where the attachment point is chlorine. Then we'll select the R2. Browse. Here I have prepared 5 acids just for demo purpose. I've used this. But if you keep chloro here, the reaction will not proceed because the reaction point is acid in this case. For example, you can see acid functionality is present in the structure. Okay, so you need to change this chloro to carboxylic acid. Okay. Once it is done, R3, R4, R5, we will not go into that now. Okay, SMRK's reactions are there. You can see here as I have shown you this in the presentation also. These many types of reactions possible or we have included in the software, but these are not limited. You can enter your own reaction here and you can save that. And you can use for the, your specific library generation. Okay, so I'll run this and we'll see how many molecules we'll get. It will take hardly 5 to 10 seconds, not more than 1 minute. Okay, the job is done. We submitted 8 haloalkanes and 5 acids. Okay, and from that we got the 40 compound library. So if you can see the 2D structures of those. You can see the reaction on the amine part. You can see the reaction on the acid part. You can see the naming pattern. You can select different naming patterns from the dialog box. 1, the first of halide reacted with the first of acid. Then first of halide reacted with the second of acid. Same up to 5 acids. Then second halide with different acids. Third halide with different acids. So in this way, you can get your library. And using Pareto method, you can optimize or prepare different sets depending on the required properties. Okay. Hope this is clear to everyone. Any doubt in this, or if you have any question, we can discuss further. Then another technique is fragment library by reaction. Okay, so for this, I will use same reaction, that is acid and amine for amide generation, just for demo purpose. You can use different types of reactions. If you don't find that reaction in the software, you can add your own reaction. That is possible. Okay, I've prepared a set of acid and amines. 45 acids are there and 35 amines are there. amide formation .rxn. As I mentioned during the presentation, we require a reaction file in .rxn format. I'll show you this file. This is simple reaction scheme, nothing difficult in this, and you can just save it in the .rxn format. You can use BioVR Draw, which is available on your system, or you can use ChemSketch, ChemDraw, anything. Okay, so I'll import these acids as well as amines in the same molecular window. Okay. So now we'll see how it works. Enumerate library by reactions. So input reactants, I will select all. There are only two visibles, but I want to carry out reactions between all these reagents. So I'll select input reaction all. Input reaction file, what we have prepared, that RxN file. You need to select that. Okay. If there is possibility of multiple reactions, then you have option use first mapping or use all types of or each mapping. That means all possible reactions. That is all option is also available. Depending on your chemistry knowledge, you can select this. I will keep it default. Okay. Then. Okay. Other things are normal. So now I will run this. Send it to background. You can see this is running. So library generation is very important technique. If you go to Zinc database, if you go to SNX or other database, you will find huge number of molecules, but all molecules may not be suitable for your project. So you need to decide which type of library you want, and on that basis, you can cluster using BioVia Discovery Studio, or you can perform your own library. Okay, so this is complete, and we used 45 acids and 35 amines, right? So it is around 80 reactants we used, and out of from those we got 75 molecules as library. So you can click on view results, and you can see 1575 molecules. Okay. Amine 1 reacted with acid 1, amine 2 reacted with acid 1. Same type of combinations will occur here. And you can see a big library is generated based on your reaction. Okay, now here two options are available with you. You can generate your own reactant library, or you can download from PubChem. In PubChem, you can select specific functionality, let's say carboxylic acid. You will get huge database. You can apply filter, and depending on your requirement, you can select specific set of the molecules. It's up to you which type of molecules you want. You can download from the huge available database as well. So in this way, you can generate your own reaction-based library using your own reactions which you are performing in your project work. Okay, now we'll discuss the novel ligand generation technique. Okay, so here we are using the breed approach. So if you have few molecules with you, you can use this breed technique where overlapping or swapping is done, and you will get big data set. And here you can see generations, up to 6 generations you can perform using Discovery Studio. Okay, so for that, first I will open a data set which I have prepared for you. Okay. I can drag and drop, I can go to file and import, I can go to file and open, all options are possible. So if you can see here, we have 48 molecules. I've downloaded around 150 factor 10A PDB structures, and depending on the biological activity of preoccupied ligand, I've screened those PDB structures, and finally I've selected, after applying 4 to 5 different filters, I've selected these 47 or 48 PDB structures. I aligned all those PDB structures and ligands were aligned in the active site, and removed all the protein structures so that I got the coordinate based alignment of different ligands. So you can see atom based alignment is not there, but coordinate based alignment is there. Okay. So these are orientations of the molecules in the receptor active site, which are downloaded from the original coordinates. Okay, so now these are 48 molecules. We'll use novel ligand technique. We'll use first generation only for demo purpose, because if we go for second or third generation, it will take more time. Okay. So input ligands, you can use selected or visible or all. We'll select all molecules here. These parameters, that maximum distance and maximum angle, will keep as it is as per the rule of from the original paper. You can see it is older technique, but very useful technique. Okay, you can go through this paper to know more about this. Okay. So we'll select all molecules. We'll keep this as it is. Generation only one. If you want to minimize the generated library, you can do that. If you don't want, you can keep it false. Or if you want to minimize, you can change it to true. But for now, I'll keep it false because it is demo purpose. If I go for minimization, then it will take more time. Okay. So all molecules, I'll run. For these 48 molecules, it will take around 3 minutes. So meanwhile, if you have any question or doubt, we can discuss on this. I'll send this to background. So if you have any other queries, so we can explore that part simultaneously.
So in library design, you select the function of the carbon and select the percentage. So on which basis you select that?
On the where you want to grow the molecule. For example, the aminophenol I've shown. There are two possible reaction centers, that is alcohol and amine. So if you want to grow on the alcohol, you can select only alcohol part, hydrogen of alcohol. If you want to grow on the amine part, you can select one of the hydrogen of amine if you want to get the secondary amines. If you want to get the tertiary amine, you can select both the hydrogens of amines. So one hydrogen will be assigned R2, another hydrogen will be assigned R3. And on that basis, you can grow the molecules or generate the library.
In the chiral section of the molecule, is it possible to integrate different enantiomers?
Enantiomer, all possible structures will be generated in that. If you use the minimization step. So after this, we'll see the recap technique and then we'll conclude our session. So I may take around 5 to 7 minutes more. What is the time? Yes.
I think your voice was really very smooth. It had a calming influence.
Was it audible to everyone? That is more important.
Please ask those who are already using Discovery Studio. Ashish, you did not show FPP. FPP.
No, sir.
Is that?
Yes, it is there. We'll discuss after this. I'll show you. Okay, so it took 2
Here's
minutes.
a question.
Yes, please.
Like we are selecting particular site, right? We are selecting particular site for happening of reaction, right? So if we select a wrong site and whether that reaction will happen or not.
If the reaction is possible by chemistry rule, then only it will continue. Otherwise, it will show error and it will mention that particular error that this is not possible.
Whether, like, what kind of error will be? Like in case of C Docker, I am getting it like ligand poses are not right. Ligand poses are not, but the ligand poses are defined, but in receptor they are getting overlapped. Like if you are having aromatic ring in your receptor and aromatic ring in your ligand, so they are getting overlapped, and so that is why that is not getting docked. So that is not the proper reason for that.
That is not getting docked.
So that is not the proper reason. So you have that enumerated thing. So here I'm asking like whether it will give the detailed error or like only one sentence error.
No, on the screen it will give a small error, but if you go to your project file, you may find the detailed error. And if you have a specific query, then you can show me your project part on your system. Then we can discuss on that in detail also with that specific error. For generalized, we cannot say this. Okay, so this is done. So I'll check the results. So from these 48 molecules using first generation approach, we got 523 ligands. And these 523 ligands are not covering these 48 molecules. So these 523 ligands are completely different from these 48 molecules. So there is no repetition in that. So we'll see the structures, we'll view results. You can see these are different factor 10 inhibitors. I have not done minimization, so some geometrical visualization error may be there. So in this way, you can go for generation of your library and then you can perform different steps on these molecules. Okay, the last technique which we will discuss in demo part is generate recap fragments. So for that also we'll use same library. Okay, the 48 molecules from factor 10A inhibition. So as per medicinal chemistry rule of 3 is followed for the fragments. So if particular fragment is following rule of 3, then you can consider it as a suitable fragment for any type of fragment based discovery technique. Okay, the fragment should be small enough that molecular weight should not be more than 300. The hydrogen bond donor should not be more than 3. Acceptor should not be more than 3. A log P should not be more than 3. Then rotatable bond, I guess, not more than 3, and polar surface area should not be more than 60 angstrom square. Okay. So this rule is there, and this is optional. You can remove any one or multiple for your study purpose. So we'll see aligned ligands. That means the name of the data set is aligned ligand. That is why it is showing here that way. So we included all the molecules. And let's see within fraction of time, it will generate or it will give you a library of fragments from these reported or known ligands. So if you are working on factor 10A, you can use these fragments to grow your own core structure. Okay. So we used 48 molecules and as per rule of 3, or on the basis of rule of 3, it has generated 57 fragments from those 48 structures. You can see these different fragments. All these fragments are having molecular weight less than 300 or whatever criteria we have selected. Okay. So I think okay, so we are done with that. For any further discussion after 2 days, if you want to further discuss, you can contact us anytime on the given email ID or contact number, or within these 2 days, or if right now, if you have any query, we can discuss. Online participants can take screenshot of this.
So please join me in thanking Thank you very much. Request Professor Coutinho to felicitate Dr. Ashish, which is in the middle, if you can.
Scientific expertise is shaping innovation and differentiation across multiple industries, including life sciences, consumer packaged goods, energy, process and utilities, industrial equipment, natural resources, high-tech, and aerospace and defense. In life sciences, Dassault Systèmes Biovia helps researchers to model individual processes within cells to identify the most promising candidates for drug research, development, and commercialization. In consumer packaged goods, Dassault Systèmes Biovia helps harness new biomaterials, smarter manufacturing processes, and innovative lifecycle assessment tools to reduce environmental impacts and move products to market faster. In energy, process, and utilities, Dassault Systèmes Biovia computational modeling and simulation tools reduce cost and increase profitability by optimizing plant engineering, construction, operation, and decommissioning. In industrial equipment, Dassault Systèmes Biovia helps manufacturers achieve disruptive innovation by optimizing the complex chemical transformations and processes that convert raw materials into functional devices, including batteries, displays, and wearable systems. In natural resources, scientists, engineers, and planners use Dassault Systèmes Biovia materials measurement, prediction, and analytics to more sustainably plan the commercial use of natural materials and Earth's resources. As the high-tech industry moves beyond Moore's law to functionally densified, highly integrated, complex designs, Dassault Systèmes Biovia is a critical enabler of polyfunctional materials design. In aerospace and defense, Dassault Systèmes Biovia modeling tools have advanced the development of strong, lightweight, and easy-to-manufacture carbon fiber composite materials for aircraft structural parts. With over 225 full-time PhD scientists on staff and an active science council, Dassault Systèmes Biovia continuously conducts innovative research across a broad spectrum of disciplines. Dassault Systèmes Biovia is committed to science. Over $1 trillion of scientific projects leverage Dassault Systèmes Biovia. Over 2,000 companies worldwide delivering scientific solutions for more than 25 years. Scientific solutions created by scientists. 110 patents generated. 38 scientific awards. 1,200 peer-reviewed publications. 29% PhD. 38% of staff have more than 10 years in science industries or scientific projects. More than 20% of staff have held university positions. Dassault Systèmes Biovia advances chemistry, biology, and materials, helping scientists innovate, invent, and create. Dassault Systèmes Biovia, scientific solutions created by scientists for scientists.
recorded आहे त्याच्यामध्ये, but I'll just say a few things. Total time नहीं पता चला. This is a pre-recorded session. 16 minutes. ठीक है.
Someone is presenting.
This is my voice. It's a recorded one. It's my voice recorded. It's a presentation, but recorded voice.
Okay.
It's fine. I'll start.
Karu? It's better to use Poland.
Yeah. I'll stand.
So sometimes this might gives problems. That's okay.
This is online record. पण मी सांगतो अगोदर ते. It is all about. अच्छा. Okay. हाँ. हाँ, 15 मिनिटाचे मी पटापट संपवते and then we'll go on. All right. Very good afternoon. I'm sure many of you are feeling sleepy, but we'll have to wait for tea. Another 15 minutes, and of course this presentation is for 15 minutes as well, around 6 minutes. It's a presentation with a recorded voice because this is what we submitted for Drug Discovery Hackathon 2020, and ICT Force was the name of our team. Now, why I want to show this presentation? First thing we have done extensive work of this, like most of the work what we have done, we use OpenAI software tools, and we also proposed a new concept called peptide mold. You will see what it is all about. But before that, let me just say a few things about me. My name is Prashant Kharkar, and I'm working as a professor of medicinal chemistry at Institute of Chemical Technology. It's been 3 years, or rather 3.5 years, that I joined ICT. Before that, I was working with NMIMS and Dr. Vaibhav and me, we were colleagues. All right. So what have we done so far? As in, I'm sure some of you might have taken a look, maybe a LinkedIn page or maybe ICT website, my profile. So it's been quite some time I have been working in this particular field, and Ashish just told about Professor Coutinho. So I always proudly mention that Professor Coutinho was my guide's first PhD student at ICT, and I was my guide's last PhD student at ICT. So he's my Guru Bandhu, and there is this very special connection. And of course, he has been the role model as far as working in this particular field is concerned. Now, having said that, just to tell you, I've been working in this field for almost 25 years. 1998, I came to ICT for my master's to work with Professor V.M. Kulkarni, and that's where I started learning these things. And whatever little I know, I have learned all these years. So I never claim that I'm an expert, and I have been on the user side. Like I have not really developed any methodology or so like you have heard of Mr. Coutinho and others. But what I do as a user, I do it very religiously, very diligently. So far, we have got 2 molecules in phase 1 clinical trials. The second phase 1 clinical trial is about to start. And it is, believe me, it's a 10 years of sheer hard work. So, of course, I haven't done it all by myself, but it is in collaboration with a company named Godavari Biorefineries Limited. So I work with them as a consultant, and it's been more than 10 years that I'm associated with them. So when I joined NMIMS around same time, I joined them as an external consultant. So they had one natural product lead, and they were trying to develop it for cancer stem cell inhibition, which is another new thought process in discovery of anti-cancer agents. So it is the cancer stem cells that those are responsible for so-called cancer relapse. So if you kill, the philosophy is if you kill the cancer stem cell, the cancer will not be back, will not relapse. Right. So with this, and currently there is no molecule which we can call it as cancer stem cell inhibitor. The highest molecule is in phase 3, but it is yet to get into clinic. So we have started working on this lead, and then we have used a variety of computational tools and medicinal chemistry intuition. And then the first patient was dosed on February 8th this year. And then during COVID, we also came up with another molecule which is viral entry inhibitor. So it doesn't kill the virus, but it inhibits the viral entry. So we believe that it will be useful for emerging viral infections. No matter if there is any pandemic in future, it will be there. Okay, now as far as this work is concerned, so what we did was we kind of came up with a concept called peptide mode. So I'll skip the initial part because I'll just describe it. So when COVID-19 started, and then a lot of structures, a lot of proteins of COVID-19 virus were proposed, or rather SARS-CoV-2 were proposed. So one of them was PL-Pro, and the other one was M-Pro or 3CL-Pro proteases, which basically process the viral protein, and initially the viral life cycle begins. So what we thought is, so you have a protease which is known to identify proteins, because the protein substrate proteins will bind and it will chop it off. Eventually the protease reaction would happen. So we thought of preparing a mold of that particular active site. Now, how do we do that? Of course, we have a lot of programs which can identify the site, and then maybe using, putting water in the site, and then there are many other technologies available. So we thought of using peptides, right? So what we did was we thought of using the tripeptide, but then tripeptides were a little smaller, pentapeptides were a little bigger. So we, in a way, settled, given the Lipinski's range of different properties. I mean, I wouldn't say all properties, but at least the molecular weight. So we settled on tetrapeptide. So we have prepared, so there are 20 amino acids, 20 into 20 into 20 into 20, 160 amino acids, that's tetrapeptides. We have a library was created. Then, so we have processed this library. So now building these structures was manually not possible. So that's where we used the OpenEye tools, where we were fortunate to, somebody has done work where the tetrapeptide smiles were deposited. So as a supplementary information, those were ready. So using one of the softwares, we converted these, well, not exactly software, as in I would say OpenEye tools, we converted these smiles into 3D structures and then processed all these 3D structures. And this is exactly what we are going to see upstairs. So after that, we generated 200 conformations of each of these tetrapeptides. So that would be 160,000 into 200. That would be around 32 million conformations we generated. All of it was done using Omega OpenEye tools. Then the next thing was to actually dog these molecules into 32 million conformations of 160,000 compounds, tetrapeptides. So then we dogged it, and then we scored all these 32 million conformations. And then we selected top 500 conformations or poses. Right, and after that we did a diversity analysis of that, and we selected top 10 best molecules along with their poses. So in a way, these combinations, like all 160,000 combinations, where that included every amino acid, all possible combinations, hydrophobic, polar, acidic, basic, and others. So then our thought process was, if you have got a tetrapeptide which is binding very nicely inside that particular active site, and then we have, like I said, we have done the diversity analysis and we have taken 10 best. So then we have taken those binding poses, and from that we derived the pharmacophore. The pharmacophore we derived using Schrodinger tool space, and then we have used that pharmacophore for screening a library of natural products, pharmacophore screening. So similarly, that natural product library, again, we got SMILES notations of these all 158,000 natural products. So then we removed those which were really big, beyond say 1,000 Daltons or so. And then we were left with 142,000 natural products. Again, 200, we processed that library, converted from SMILES to 3D, and then charges and everything. And then eventually again processed their conformations and used those conformations for the pharmacophore matching. And at the end of it, again, so then we got this pharmacophore hits. And then those hits were again docked back inside the active site of this protein. And then eventually, so we put all this thing together and we came up with 10 best molecules, because the challenge was to identify natural products which would be, very potently or very, with great affinity they would bind to the active site. Then you may ask, we would have simply docked the molecules and we would have got. So then we also did that, and then we could see there was a very minor overlap. Then so at the end of it, so the idea was, it's like you have an active site, so you put clay on that, so you get an imprint of that active site. That's exactly what we did. We docked the molecules, we got that imprint and used that imprint for creating molecules which were similar to that. So this is exactly what was done in this particular presentation. So I will just quickly go through this. Hopefully, of course, I have described the methodology. We haven't really published this paper, which we have communicated. Yeah, so what was more important is, of course, this was a new concept, and since although this was for proteases, but any protein target that we deal with, this concept can actually be used for coming up with novel chemistry. Because ultimately, when you work in drug discovery, the aim should be to be able to come up with novel chemistry. Because if that chemistry is not novel, then course, you will not get any intellectual property associated with that. So to be able to be a very good modeler, you need to be very thorough with the chemistry, organic chemistry, the functional groups. If you do not understand chemistry well, then you should make some conscious effort, because if you don't do that, then everything is Greek and Latin. Like in the morning, Shastri said that for many people this is like a black box. They know how to use the software, but then once you know how to use the software, but what about the idea generation? Right? And this is where you should fall in love with the structures. And once you do, you can actually really excel in what you do. That is drug design part. Otherwise, these softwares are there, but ultimately one person and meaning two modelers, they will differ from each other in their ability to generate newer ideas, right? And then, of course, for all of this, you need to read a lot, and you need to also take a look at newer tools and newer technologies that are introduced, because again, like Sir mentioned, these softwares are going to change over a period of time. They will not be same, but the technologies or the basic philosophy is, and this is where understanding how these softwares work is very important when it comes to, using software is one thing, but then understanding the philosophy behind what you are doing, why you are doing, that's very important, right? So most of the work was done using OpenAI software tools. And just so you know, I do not represent OpenAI. Just that I know I have used OpenAI for very effectively for many different things, particularly the ligand-based drug design. I'm a very big proponent of ligand-based design because when you talk about structure-based drug design, what we do, molecular docking, is an oversimplification of what actually happens at the biological or molecular level. So, I mean, sir, everyone would agree, sir would also agree, if you can have a good correlation between the binding, meaning the actual experimental value, and the docking score, would be very lucky. And those are the times which are very rare to find, right? So again, what I'm trying to tell you is these are all tools, and how effectively you use them, it all depends on how you generate your ideas and how you make things work in your favor, right? So don't do just run of the mill kind of work. Come up with something interesting, and believe me, the more you read, the better you get at generating ideas. So everything revolves around generating newer ideas. Okay, so let's take a break, and of course there is another small presentation, but I think that's related to OpenAI, so we'll do it when we go up. Okay.
We take this opportunity to thank Professor Kharkar.
The job is not over yet.
Yes, but we can already felicitate you. Sir, please,
I
request Professor Coutinho to.
Another fortunate guy in this going to get felicitated by sir.